Hello everyone, very warm welcome to Sri Chaitanya Academy. My name is Kiran Chaudhary and I will be discussing your today's grand test papers botany questions. So let's get started guys. First question on your screen. It was a match list 1 with list 2. Very simple from your Cell the Unit of Life chapter. See, the first one is Pili. Pili is going to match with elongated tubular structure made up of special protein. So, pili is 4. Fimbriae. Fimbriae is small bristle-like fibers sprouting out of the cell. Mesosomes, specialized and differentiate form of cell membrane. Very simple one. Chromatophores will contain pigments. Very simple. Match the column from your Cell the Unit of Life chapter and the topic is prokaryotic cell. Very simple. 4, 3, 1, 2. So it should be 4, 3, 4, 3, 1 and 2. So 4 should be your right answer. 4, 3, 1 and 2. 4, 3, 1 and 2. So 4 is your right answer. Correct guys? Next question. Very simple. Which of the following best describes the role of oxygen in aerobic respiration? Please focus on the terms guys. Everyone present in this class. So oxygen leads to activation of glycolytic enzymes? No. Oxygen is the driving force for withdrawal of hydrogens from pyruvic acid in stepwise manner. This is in fact, my dear students, correct. And oxygen acts as a final hydrogen acceptor in aerobic respiration. Even this is correct. So A is incorrect, but B and C is correct. So you were supposed to find out best describes means the statements which are correct over here with respect to aerobic respiration. So B is correct and C is correct. So answer is B and C only. So your correct answer is, my dear students, 4. B and C only. Simple? Good. Next. Different types of alcoholic drinks are obtained based on type of raw material used for fermentation. Yes, absolutely correct. Type of processing, yes. With distillation, without distillation, likewise. Type of microbes involved, no. This is purely from your NCERT. A is correct and B is correct. So if you see over here, A and B only. So my dear students, 1 becomes your absolutely correct answer. A is correct and B is correct. C is incorrect. So answer is 1. A and B only. Simple. Everyone present in this class, give me a thumbs up if you're listening guys, and please participate. Let me know which particular answer, which particular option you have given as answer. Please participate. It will become a revision for you again. Next question. Match list 1 with list 2. Lot of match the following. Citric acid, Aspergillus niger. Second. Streptokinase from Streptococcus. 5. Cyclosporin A, Trichoderma polysporum. Statin you are going to get from Monascus. So 2, 5, 1 and 3. So 2, 5, 1 and 3. So 1 is your correct answer. Next question guys. Oogamous type of sexual reproduction can be seen in very simple out of the 4. Volvox becomes your right answer. Answer is 3 guys. Volvox. Next question. Statement 1 and statement 2, you have to read and find out the correct answer from the given option. The carbon skeleton produced during respiration is used as precursor for biosynthesis of other molecules in a cell. This is absolutely correct. Plants unlike animals have no specialized organs for gaseous exchange, but they have stomata and lenticels for this purpose. This is also from your NCERT and absolutely correct. So the right will be answer from the above is statement 1 and statement 2 both are correct. So both statement 1 and statement 2 are correct. So 2 should be your right answer. Okay guys, come on, next question. Select the incorrect match. Formation of bivalence. Does it take place in zygote? Yes. Crossing over takes place in packetine? Yes. Appearance of chiasmata, the egg-shaped structures, diplotin. This is also correct. Segregation of factors in metaphase 1. This is incorrect and hence this becomes the answer. So my dear students, the correct answer for this question is 4 because they are asking select the incorrect match. Select the incorrect match. Answer is 4. Clear? Very good. Next. Fluidity. Fluidity of membrane is measured as, this is again from your NCERT, right? Cell, the unit of life, eukaryotic cell, from your cell membrane topic, where they have mentioned about fluid mosaic model. This is also directly from your NCERT. Fluidity of membrane is measured as lateral movement of proteins within the overall bilayer. So answer is 1. Clear? Give me a thumbs up if this much is clear. Next. The tail of functional mRNA is made up of poly A. Very simple. Very simple question. Next. Statement 1 and statement 2. Okay. Each photosystem has reaction center and light harvesting complex. Correct. Light harvesting complex helps to absorb lights of various wavelength and reaction center is involved in photo excitement of electrons for ETC. That is also correct. So both the statement 1 and statement 2 are correct. So both statement 1 and statement 2 are correct. So 1 will be your answer. 1 will be your answer. Clear guys? Excellent. Next. Theoretically, the modified alleles could be responsible for the production of normal or less efficient enzyme, yes. Non-functional enzyme, yes. No enzyme, this can also be the condition. So depending upon what kind of mutation it was. So either the normal or less efficient enzyme, a non-functional enzyme, or no enzyme at all. So A, B, C all are correct. Answer is 4, A, B and C. Differentiation in plants is also open like that of growth. Correct statement. Cells or tissues arising out of the same meristem have different structures at maturity. This is also correct and very correctly the reason is explaining the assertion statement. So both assertion and reason are true and reason is the correct explanation of assertion. Answer is 1, my dear students. Very, very simple and interesting. A good question. Next, alteration of DNA sequence, that means change in the sequence of DNA, in the nucleotide sequence of the DNA, and consequently resulting in change in genotype and phenotype of an organism. So which of the 3 is capable of changing the sequence, nucleotide sequence in the DNA? So if you see, yes, mutation is capable. Crossing over, yes, crossing over is also capable. But independent assortment, no. Because the question is about change or alteration of DNA sequence. So the term is very important. So A is correct, B is correct, but C is not correct. So answer is A and B only. So your correct answer is 1. Out of the 4 options, answer is 1. Clear, guys? Excellent, excellent. Give me a thumbs up, all those who are listening. Next, read the following statements. DNA is a polymer of nucleotide. This is correct. In RNA, every nucleotide residue has an additional OH group present at second position of ribose sugar. This is also correct. Backbone of polynucleotide chain is formed due to sugar and phosphate. Correct. A typical nucleosome contains 200 base pairs of DNA helix. This is also correct. So A, B, C, D, all are correct, guys. You're supposed to choose the correct statements. So A, B, C, and D. So 3 becomes your right answer. Clear, everyone? Next. Avery and his co-associates observed that proteases and RNases did not affect transformation unlike DNAs. This is correct. Transforming substance, that means the transforming principle, was not protein or RNA. Instead, it was DNA. Yes, and that is why DNAs was the one that was affecting transformation. So, absolutely correct, and reason is also correctly explaining the assertion. So, both assertion and reason are true, and reason is the correct explanation of the assertion. Answer is 1, my dear students. Next on your screen. Read the following statements and pick the incorrect one. Pick the incorrect one. So, if you see over here, rest all are correct, but the incorrect one is the second one here. Only DNA has the ability to generate its replica. No, even RNA can also replicate can create its copy, okay, through complementary base pairing rule. So only DNA has the ability to generate its replica. This is an incorrect statement. Incorrect statement. Answer is 2. Next guys, answer is 2. Xylem component tracheids are present in. So tracheids will be there in, or xylem component tracheid will be there in pteridophytes, gymnosperms and angiosperm. So B, D and E. B, D and E only. Answer is 4. Clear? Yeah. Let me know if there are any doubts in any of the question or option explained so far. Answer 4. Match again. SER, smooth endoplasmic reticulum, is involved in glycogen metabolism. 3. Golgi body, formation of glycolipid. 1. Cytoskeleton involved in motility. 2. Rough endoplasmic reticulum RER is involved in protein synthesis. Is 4. So 3, 1, 2, 4. 3, 1, 2 and 4. So 3 should be your right answer. Next. Okay, again AR. Eukaryotic cells perform cellular functions with high efficiency. Correct. This efficiency is achieved through compartmentalization, where membrane-bound organelles such as nucleus and mitochondria allow distinct cellular processes like DNA storage and energy production to occur in optimized environment. Correct. Yes, and the reason is also explaining the assertion. So both assertion and reason are true, and reason is the correct explanation of the assertion. Answer is 1, my dear students. Your correct answer is 1. Next, very good. Meiosis II resembles a normal mitosis. This is correct. In anaphase 2, there is splitting of centromere. This is also correct. In anaphase 2, there is splitting of centromere. So, both the statement 1 and 2 are true. So, both the statement 1 and statement 2 are correct. Answer is 3. Next, guys. Okay, interesting question. How many DNA molecules will be radioactive in metaphase if a human cell with no radioactivity in G1 phase is cultured in radioactively labeled thymine containing medium? So there are 2-3 things you need to notice here. They are starting with a human cell. So you can consider a diploid human cell. So how many chromosomes are there in a diploid human cell? 46 chromosomes. So you can consider 46 DNA molecules. Now, notice here, they are asking how many DNA molecules will be radioactive. Now see over here, my lovely kids, it is starting from a normal. That means the cell which is entering into the cell cycle, it is having a normal DNA. Only when the DNA is going to get replicated, when the DNA replication will take place in the S phase. And please notice, please remember that DNA replication is semi-conservative in nature. That means every new DNA molecule that will be synthesized, it will have one parental strand, that means normal in this case, and one new strand, that means radioactive. So every DNA molecule is going to have one normal and one radioactive, right? So overall, making the DNA molecule radioactive. So all the DNA molecules will be radioactive. So DNA replication is happening, right? So they are asking about metaphase. So yes, the DNA would have been replicated in the S phase. So please remember G1, S, G2 and then M phase start. Okay, M phase is going to start with prophase followed by metaphase, anaphase, telophase. Then at the telophase, your karyokinesis is going to finish and then cytokinesis. At that, after the cytokinesis, what is going to happen? You will get the new daughter cells. So please notice, since they are asking about metaphase, which is after S phase, so those 46 will be replicated. Now you have 92. So from 46 double, 92 DNA molecules will be radioactive. The reason is DNA replicates semi-conservatively, in short. Clear, guys? Next. So the correct answer is 2. The correct answer is 2. 92. Answer is 2. Next, everyone. Match. Cyanobacteria. Cyanobacteria is a blue-green algae. 1, 2, 3. Okay. Golden algae is desmid 1. Eubacteria is a true bacteria 4. And Phycomycetes albugo. So it is 3, 1, 4, 2. 3, 1, 4, 2. So 4 should be your right answer, guys. 4 should be your right answer. Next. Ray and disc florets can be seen in, absolutely simple, sunflower. Your correct answer is 1, sunflower. Next question. Chemo, right? Please notice chemo. Chemotaxonomy is based on chemical constituents of a plant. Answer is 2, my lovely kids. Chemotaxonomy is based on chemical constituents of a plant. Answer is 2. Next, everyone. Which of the following is a product of de-differentiation? Very simple. Answer is 3. Corcambium. Which group of organisms belongs to Basidiomycetes? So if you see over here, mushrooms, Astilago and Pyxenia. Answer is 3. So the correct answer is 3. Mushroom, Astilago and Pyxenia. So answer is 3. These all belong to Basidiomycetes. Which of the following is a growth inhibitor? Growth inhibitor, obviously, it has to be the abscisic acid. Answer is 2. Abscisic acid. Very simple. Growth inhibitor, abscisic acid. Answer is 2. Next. Okay. Match list 1 with list 2. So A, UAG. So UAG is the stop codon. RNA with introns and exons is the hnRNA 3. RNA with exons only is your mature RNA 1. Nucleosomes, if I talk about, so nucleosomes you will see in chromatin structure or chromatin form of DNA. So 4, 3, 1, 2. 4, 3, no, it is not matching. 4, 3, 1 and 2. So 1 should be your right answer. Okay guys, 4, 3, 1 and 2. So 1 is your correct answer. Next. Statement 1 and statement 2. Okay, again, streptokinase is produced from a fungus. No, it is produced from a bacteria, streptococcus. So this is wrong. Statin is used as an immunosuppressive agent. No, this is also wrong. Statin is your blood cholesterol lowering agent. So both statement 1 and statement 2 are incorrect. So answer is 4 guys. Answer is 4. Both statement 1 and statement 2 are incorrect. Next question on your screen. How many, very simple, how many ATP molecules are gained from one glucose? How many ATP molecules are gained from one glucose during fermentation? So answer is 2. 2 ATP. Your correct answer is 2 ATP. Next. Which one of the following organism is correctly matched with its 3 characteristics? So if you see over here, the fourth option, maize is a C4 plant and maize is a monocot. So it will have closed vascular bundles and scutellum also in the seed. So correct answer is 4. Right guys? Question number 123. Correct answer is 4. Next. How many of the given plants have actinomorphic flower? Very simple from your very basic NCERT you can follow for that. But please notice, if you are following the latest NCERT, you might not find a few example. But basically, if you follow in plant families, if you follow, my dear students, the Brassicaceae family, if you follow Solanaceae family, Liliaceae family, Leguminosae family, and also along with the discussion which is about the flower, and the symmetry of flower, which is given also in your NCERT, the discussion on flower before the families. So if you have gone through all this, this question is very, very simple. So if I see how many are here, total 12 are here. So radish, yes. Tulip, yes. Ashwagandha, yes. Actinomorphic. Lupin, no. Cassia, no. Canna, no. Asparagus, yes. Lily, yes. Datura, mustard, chili and Tobago. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Yes guys? So if you see over here, the correct answer is 9. So you have to leave lupin, you have to leave cassia, and you have to also leave canna. So 1 lupin, 2 cassia and 3 canna. So total how many? 9 are left. Answer is 2, 9. Clear? Next question. both asexual spores, listen this carefully, both asexual spores and gametes are non-motile in red algae. So your correct answer is 4, red algae. Next question on your screen. Which of the following is not associated with eukaryotic flagella or cilia? 9+2 arrangement of microtubules, correct. Naked and not covered by plasma membrane. This is the correct answer for this question because this is an incorrect statement. Emerge from basal body, correct. Exoneme of tubulin protein, this is also correct. So they are asking about which of the following is not associated, that means incorrect. So your second statement which says that the cilia flagella are naked and not covered by plasma membrane, this is incorrect, it is not associated, hence it becomes the answer to this question. So answer is 2. Okay, yeah, answer is 2. Select the incorrect pair. So if you see over here, the third option, and this is the incorrect pair because Joseph Presley, role of air in growth of green plants is correct. Engine horse, essentiality of light, this is also correct. Van Niel, photosynthesis in bacteria, this is also correct. So your answer is 3, my dear students. So the third one is the incorrect pair, 3. Next. Which of the following statement is true regarding ABA or abscisic acid? So if you see, you are supposed to find out the true or correct statement. ABA promotes seed germination? No. ABA is known for its role in stimulating plant growth? No. ABA aids in closing stomata and helps plants to tolerate the stress. Yeah, that is correct. So this is the true statement regarding ABA or abscisic acid. ABA primarily functions as catalyst? No. So answer is 3. Answer is 3, guys. Okay? Yes, very good. Next. Which of the following is a structure or a type of asexual reproduction that mimics sexual reproduction? It has to be your apomixis. Answer is 3. Apomixis. Clear, guys, everyone? Excellent. Next. What is the ploidy of embryo sac of angiosperm? So it is, my dear students, haploid. Answer is 1N, or you can say haploid. Clear? Very good. Next. Select the correct match. Select the correct match. So see over here. Monocot stem open, no. Dicot stem closed vascular, no. Stem radial, no. Fourth, conjoined vascular bundles will have phloem located on the outside of the xylem. So yes, this is the correct match. Answer is 4. You were supposed to find out, my dear students, correct match. You were supposed to find out correct match. Answer is 4. Good. Next. Next question on your screen. In Kelvin cycle, for the synthesis of one glucose molecule, in Kelvin cycle, for the synthesis of one glucose molecule, how many ATP and NADPH plus H plus molecules are needed in total? Very simple, straight from your NCERT. They are asking about one glucose molecule in Kelvin cycle. So obviously the answer is going to be 18 ATP and 12 NADPH plus H plus. So your answer is 2. Your answer is, my dear students, 2. 18 ATP and 12 NADP H plus H plus. Answer is 2. Clear? Yes, everyone. Next. So match the plant growth regulators in list 1 with their corresponding discoveries or effect in list 2. So auxin isolated by F.W. Wendt from oat seedling of coleoptile tips. So it should be 3. Gibberellic acid identified from the Botany disease in rice seedling 1. Kinetin promotes cytokinesis in plants or plant cells. Abscisic acid or ABA, chemically identical to inhibitor B abscission 2 or dormine. This is 5. Ethylene is identified as a gaseous PGR that hastens the fruit ripening. 4. So if you see over here, so 3, 1, 2, 5, 4. 3, 1, 2, 5, 4. 3, 1, 2, 5 and 4. So 2 bacha, my lovely kids. 2 is the correct answer. 2 is the correct answer. 3, 1, 2, 5, 4. So in the 2 options, 3, 1, 2, 5 and 4 is given. So 2 is your right answer. 2. Clear? Let me know if there is any doubt. Now we'll move to the next question. Mendelian monohybrid F2 phenotype ratio. Very simple. You cannot expect a simpler than this question in this particular topic or chapter. So Mendelian monohybrid F2 phenotype ratio is 3:1. That means 3 out of the 4 will be tall and 1 out of the 4 will be dwarf. So 3:1 is the phenotype ratio of F2 generation in Mendel's monohybrid cross. Answer is 2. Yes guys, answer is 2, 3:1. Answer is 2, 3:1. 2. Clear? Yes. Next. Statement 1. Competition affects carnivores more adversely than herbivores and producers. This is wrong. Now you must be knowing this very nicely, okay? This has been getting repeated again and again. So, competition affects herbivores and producers more adversely than carnivore. This is the right statement from your NCERT. According to Gauss, 2 closely related species competing for the same resource cannot coexist indefinitely. So this is correct. So statement 1 is incorrect, but statement 2 is correct. So answer is 4. Answer is 4. Answer is 4. Okay, next. VNTRs are usually coding DNA? No. Non-inheritable DNA? No. Repetitive sequences whose copy number remains constant across all chromosomes in all individual? No. So VNTRs is a mini satellite. That is right. So yes, this becomes the right answer for your question. VNTR, variable number tandem repeats, are a part of mini satellite DNA. Answer is 4. Clear guys? Next. Which of the following is one of the reasons that contributes to greater biological diversity in the tropics? Relatively stable and undisturbed climatic condition. Answer is 2. Next. The interaction between which of the following pair of organisms is classified as commensalism? So if you see barnacles which are growing on whale, this is commensalism. Answer is 2. Next. Which of the following is inverted? Very simple, very simple from your NCERT. So pyramid of biomass in the sea ecosystem. Your correct answer is 3. So which of the following is inverted? Pyramid of biomass in the sea ecosystem is inverted. So your correct answer, my lovely kids, is 3. Your correct answer is 3. Select the correct match. Select the correct match. So if you see over here, the second option, if you see over here, my dear students, the second option, Klinefelter syndrome, 44 autosomes, 2X and 1Y. This is the correct match. How many total chromosomes are there in Klinefelter syndrome's individual? There will be 47. So 44 autosomes, 2X additional copy of X chromosome. So 2X and Y. So 44, 45, 46, 47. So the correct answer is 2. The correct answer is 2. Yes, very good. Frishti and Nagababu. Very good. Next question on your screen. Very simple. Pedigree. So if you check out, my dear students, you will see this pedigree belongs to an autosomal dominant trait. Example, myotonic dystrophy. This is one of the very simple pedigree. So your correct answer is 4, my dear students. Autosomal dominant is the trait which is being shown over here, which we are studying here. And the example of autosomal dominant trait is myotonic dystrophy. So your correct answer is 4. Yes, very good. Your correct answer is 4. Nile perch and cichlid fishes had undergone co-extinction as both species shared an obligatory relationship. This is wrong. This is actually an example of alien species invasion. So this is wrong. Amazon rainforest is being cut and cleared for cultivating soya beans or for conservation to grasslands for raising beef cattle. This is correct. So statement 1 is incorrect, but statement 2 is correct. So your answer is 4, bachas, because statement 1 is incorrect, but statement 2 is correct. Answer is 4. Clear? Next. Which of the following organisms does the normal, in which of the following organism does the normal female produces ova carrying different types of allosomes? So this is simply a way to ask that in which of the following organism female will be heterogametic. So female heterogamety. So it will be in pigeon. Answer is 3. Your correct answer, my lovely kids over here, is 3, pigeon. Next. In his analogy of the rivet popper hypothesis, Paul Ehrlich suggested that loss of key species poses a major threat to the stability of an ecosystem. So the correct answer is 3. Yes, guys. Next, correct answer is 3. Amount of energy captured by plants to sustain the entire living world is just 2 to 10% of PAR. So your correct answer is 3, my dear students. Your correct answer is 3. 2 to 10% of PAR. So 180 question. Answer is 3, 2 to 10% of PAR. So that's all, my dear students, from botany. Now, zoology questions will be discussed. Bye-bye. Take care. All the best for your upcoming NEET exam.
Thank you. Charging ఎక్కువ రికార్డింగ్ ఆన్ పెట్టిండ్రి ఇప్పుడు ఓకే బ్లూ డాట్ వచ్చేది కదా
Hello, very good evening everyone. Hope you all are doing fine. Yes, Vrishti, Nagbabu, everyone, Girija. So this is Dr. Priyanka Day and now it's time to discuss questions of zoology. So are you ready for it? Yes, good evening, good evening. So let's see what is the first question on your screen. Okay. So this is the first question. Let's discuss it. Read the following statement. So this is a question from RNA interference. Okay. So we need to read all the statements and then we need to find out the correct statements. RNA interference is a method of gene silencing at the translational level. Yes. When we are saying RNA interference, definitely RNA is silenced. This is right. RNA interference is a cellular defense method in all the eukaryotes. Definitely it is seen in eukaryotes. RNA interference involves silencing of mRNA by complementary double-stranded RNA formation. Yes. And source of complementary RNA for RNA interference could be RNA genome of virus or transposable elements which are termed as transposons that replicate via RNA intermediate. Yes. So basically all the statements are correct regarding it. Yes, definitely. So the correct answer will be A, B, C, D, that is 4. Yes, very good. Next, when you see match the following, so you will have to match list 1 with list 2. Okay. When we talk about elution, it is extraction of DNA from the agarose gel. Okay, ampicillin resistant gene is the selectable marker. TI plasmid is a cloning vector. Restriction enzyme, obtaining DNA fragments with sticky ends. Okay, so A is matched with 4 and B is matched with 3 and C is matched with 1. Okay, so we have just one option, that means 3. Very good. Saksham, Vrishti, Nagbabu, very nice. Next. Now check this reaction and you will have to tell me this is because of which enzyme, which class of enzyme. So we have options oxidoreductase, hydrolases, lyase and ligase. Definitely there is breakdown of bond. So it will be lyases. Okay, so correct answer is 3. Yes. Choose the correct statement. All bird possesses pneumatic bone and can fly. So not all can fly. There are flightless birds as well. Isn't it? All annelids exhibit internal fertilization. Remember that in annelida, they are saying that there will be internal fertilization. So that is correct. All amphibian possess 2 pairs of limbs. So all amphibians will have limb. It's not true, right? Here it is written all annelids exhibit internal fertilization, which will be wrong. All fishes are poikilotham, which is right because fishes are cold-blooded animals. So correct answer here will be 4 because we want the correct statement. Pay attention to all the terms, all or mostly, that is also important. Okay, so it is 4. Next, genetic drift, change in allele frequency by chance. By chance is the keyword here. Saltation is single step large mutation. Gene flow is related to gene migration and inheritable factors will be matched with influence of phenotype. So D is matched with 1 and A is matched with 2. So we got our answer. It should be 2. Very good, very good. Keep it up. Samruddhi. Next. Select the option that represent the correct sequence of cross-bridge formation and its breakdown. So if you read all the statements, you will see that there is binding of new ATP, cross-bridge break, hydrolysis of ATP by the ATPase activity, cross-bridge formation and sliding of the actin filament. So I think at the end there should be sliding of the actin filament. Yes, E. And in the beginning we have 2 options, B and C. So cross-bridge breaks or hydrolysis of ATP by the ATPase activity. Okay, so this will be the hydrolysis of ATP, right? Now binding of new ATP with the myosin head. Okay, so first we will go with hydrolysis of ATP by the ATPase activity. This will be first. After hydrolysis there is a release of energy and because of which there will be formation of cross bridge. So this will be 2. After cross bridge formation there will be sliding of actin filament. So this is 3. Now then there will be binding of new ATP with the myosin head. Okay, so this is 4, and then there will be the breaking of cross bridge. So after new ATP binds, then the cross bridge will break. So this will be the last one, right? So the correct answer should be 1, right? Next, choose the correct statement from the following. Amniocentesis is not useful in detecting the chromosomal disorder. The main function of amniocentesis will be detecting the chromosomal disorder. So this is wrong. Saheli is a steroid. No, it is a non-steroidal oral contraceptive pill. A surrogate mother is always the biological mother of the newborn, right? And gonorrhea can be cured. This is wrong. And the gonorrhea can be cured completely if detected early and treated properly. So when we talk about gonorrhea, it is caused by bacteria Neisseria gonorrhea, right? And this, if detected earlier, can be treated with antibiotics. So this is true. Coming to the next question. Study the following statement regarding the frog and select the correct option. It drinks water using bilobed tongue? No. It will absorb water through the skin. Gaseous exchange occur through the skin both in water and on land. This is right because we know in frog all the 3 modes of respiration will take place on land. In female frog, the oviduct open directly to the exterior instead of opening into the cloaca. No, it will open into the cloaca. And leukocytes as well as RBCs are nucleated, RBCs are not. So correct option we have here 1 is B. Okay. So let's see, because we have 2 options here. It drinks water using its bilobed tongue, which is absolutely wrong. In female frog, the oviduct opened directly to the exterior instead of opening into the cloaca. And leukocytes as well as RBCs are nucleated. So which option you guys have marked, right? So correct option given here will be 2, B and D. So we know that the leukocytes are nucleated but RBCs are not. Okay, and we need to find out the correct option anyways. Okay, so we have this option only for now, that is 2, right? Next, acha, in case of frog, yes, definitely in case of frog the RBCs will be nucleated. Yes, so this is right. So correct answer is 2. Next, in human female, the secondary oocyte completes meiosis 2 when the sperm will enter the cytoplasm. Okay, so when the sperm enter the cytoplasm of the ovum, that means it is 3. Yes, frog mein hai nucleated, absolutely. Consider the following feature of IUDs. Make the uterus unsuitable for implantation. Make the cervix hostile to sperm by altering the quality of the cervical mucus, and inserted by doctor expert nurses into uterus through vagina. So all these will be the characteristic of hormone releasing IUD, that is LNG20. So I hope you guys have marked 3rd option, that is LNG20. Yes, very good. Next. In human beings, the brain stem comprises of, so brain stem is made up of midbrain, pons and medulla oblongata. So yes, correct answer is 4. Select the incorrect match among the following. Physical barrier is skin, physiological barrier is saliva, cellular barrier is macrophage, and cytokine barrier will not be polymorphonuclear leukocyte, it is a neutrophil. So it should be interferons. So correct answer should be 4. Right? Good. When semilunar valve of heart, so here we are talking about heart sounds. So only 2 heart sounds are audible. One is due to the closure of AV valves. Second one is because of closure of the semilunar valve. So when semilunar valve close, the second heart sound is produced. So correct answer here will be 3. Okay. In humans, plasmodium reaches the liver through blood as sporozoites. The parasite in the hepatocytes reproduce asexually. So it should be sporozoites and asexually. That means the correct answer should be 4. Okay. Now we have assertion and reason statement. Let's read it. When a single-stranded DNA or RNA tagged with the radioactive molecule or probe is allowed to hybridize to its complementary DNA in a clone of cells followed by detection using autoradiography, the clone having the mutated gene will not appear on the photographic film. This is absolutely correct, and this is how we will do autoradiography. Okay. Now the probe will not have complementarity with the mutated gene. Yes, and the probe had complementarity with the normal gene, but if it is mutated, the probe will not be able to bind it. So assertion is true, reason is true. Okay, and in fact reason is the correct explanation of assertion. Okay, so answer should be 1. Okay, so answer should be 1, everyone. So it's not 4, it's 1. Okay. Now placenta. Now placenta is a structural and functional unit between the fetus and the maternal blood. Chorionic villi is a finger-like projection. Stem cells will be present in the inner cell mass. As we know, stem cells, they form the embryo and the fetus, and the implantation will be of blastocyst. Okay. So let's see. A is matched with 3, B is matched with 1. Okay. So we got our answer. It should be 1. Which of the following arthropoda is an important gregarious pest? So gregarious pest here will be Locusta. In cockroach, uric acid is synthesized in. So we have 4 options: hepatic ceca, hindgut, hemolymph and Malpighian tubule. The answer will be Malpighian tubules, right? Statement 1 says all tetrapoda are gnathostomes, but not all gnathostomes are tetrapoda. So if we see the classification, how it goes, so we know that the chordata, okay, will be divided into avertebrata and vertebrata, right? Now this vertebrata is further classified as agnatha and gnathostomata, right? Now the gnathostomata are further classified as Pisces and tetrapoda, right? So if we read the statement that all tetrapoda are gnathostomata, but all gnathostomata will not be tetrapoda, okay, so that is correct. All gnathostomes are vertebrates. All gnathostomes are vertebrates, but not all vertebrates will be gnathostomes. So this is also correct. Both the statements are correct. So the correct answer will be 1. Okay, everyone? Yes, so it is 1. Next. Fibrinogen will help in coagulation of blood. Albumin will help in the osmotic balance. Heparin is an anticoagulant. And globulin helps in the defense mechanism of the body. So A is matched with 1, B with 3. Okay, so we got our answer. It should be 4. Next. Select the correct match. Patella covers the elbow joint dorsally. Now patella is the kneecap. Acetabulum articulate with the head of the tibia, no with the femur. Glenoid cavity accommodate the head of humerus, absolutely. And acromion articulates with the head of the femur, no. So correct option here was 3. Yes. Now good humor hypothesis of the health was disapproved by. Let's see. The discovery of blood circulation by William Harvey, yes. The demonstration of normal blood temperature in person with black bile using a thermometer. So here it is written normal blood temperature, which is right. The discovery of blood groups, no. Demonstration of high body temperature in person with black bile, no. So it should be A and B. That means the correct answer is 1. Yes, Saksham, Nagbabu, Vrishti, very good. So we have again assertion and reason question. Pelagic fishes produce small number of small sized offspring. So this is wrong. They produce a large number of small sized offsprings. Under a certain set of selection, pressure, organism evolved towards the most efficient reproductive strategy. Yes. So assertion is false and reason is true. Answer should be 3. Okay, answer should be 3. Now oxytocin. Okay, so it is responsible for milk ejection. Melatonin. Okay, so melatonin, we have here influences metabolism, pigmentation and our defense capability. Thyroxine. Okay, so thyroxine will control the metabolism, and cortisol maintain the cardiovascular system as well as the kidney function. Okay, so correct answer, that's why will be A is matched with 3, B is matched with 4. Okay, that means 2. Yes. Next question. So we need to find out here the incorrect statement. A says yellowish fluid colostrum has abundant antibodies IgA to protect the infant. So IgA is a secretory antibody present in all our bodily secretions. So definitely it is correct. Injection of preformed antibodies against snake venom is called passive immunization. Yes. The use of drug like antihistamine, adrenaline and steroid quickly reduce the symptom of allergy. Absolutely. The primary lymphoid organs provide site for the interaction of lymphocyte. No. Primary lymphoid organ help in the maturation and production, okay, of lymphocytes. To determine the cause of allergy, the patient is usually exposed to high dose of possible allergen, no, low doses. So we have A, B and C as the correct answer. Okay, A, B and C. Okay, so do you have any option here? A, B and D it is written. Correct answer is yeah. So we want here the incorrect one. So definitely we have D incorrect and E incorrect. So our answer should be 2. Okay. Next. So we have FSH interacts with the membrane bound receptor but does not enter the target cell. Yes. So FSH is a protein or a glycoprotein hormone secreted by anterior pituitary gland and it binds to the membrane receptor. Now FSH primarily regulate the gene expression or chromosome function in the genome. No, this is wrong. Because FSH is not able to enter the cell, it will bind to the membrane receptor and it will form secondary messenger. It will not help in regulation of the gene expression. So assertion is true but reason is false. So answer should be 4. Okay, answer should be 4. Select the incorrect match with respect to human excretory system. Peritubular capillaries formed by the fine branches of the afferent arteriole. Malpighian body is glomerulus plus Bowman's capsule. Yes. JG apparatus is a cellular modification in the afferent arteriole and the DCT at the site of their contact. And counter current mechanism is because of loop of Henle and vasa recta. What is wrong here is 1. Okay, so correct answer should be 1. Okay. Now let's see and read this question. An epithelium with a single layer of tall slender cell with the nucleus located at their bases is found in. So they are asking about tall columnar cells that you will get in the lining of stomach and intestine. That's it. So correct answer should be 4. Now after puberty, the human males, the dash present on the inside wall of the seminiferous tubule multiply by mitosis and increase in number. So we have here Sertoli cells which is outside, Sertoli cells which is inside the seminiferous tubule, but definitely they are not going to proliferate and form sperms. Leydig cells are outside the seminiferous tubules. Spermatids, no, so it is spermatogonia that lines the seminiferous tubule and proliferate further to form sperms. Next. Now, in a population that is in Hardy-Weinberg equilibrium, 190 individuals out of 1000 individuals show the dominant phenotype for a character. Then what will be the frequency of heterozygous for this character? So, for heterozygous, we know it is 2PQ, okay, and we know that how many individuals out of 1000 are dominant? 190. Okay, so when we will do 2 into PQ, right, we have here 1000 minus 190 will be the number of individuals having recessive phenotype, right, and 190 have the dominant phenotype. Okay, so we can calculate the heterozygous individuals, frequency of heterozygous individuals by 2PQ, which will be around 0.18. Okay, so correct answer should be 2. Next one. Vital capacity of the lungs will be. So vital capacity will include every volume apart from the residual volume. So residual volume is not included, so it will be the tidal volume, IRV and ERV. That means the correct answer should be 3. Right? Next. Which of the following correctly describes the similarity between ZIFT and IUT? So ZIFT is zygote intrafallopian transfer and IUT is intrauterine transfer. The major difference between it will be that in ZIFT, the embryo or the zygote less than 8 blastomere stage will be introduced into the fallopian tube. But in case of intrauterine transfer, the embryo with more than 8 blastomere stage will be introduced into the uterus. So transfer of an ovum collected, no, there will be never a transfer of ovum in ZIFT and IUT. Fertilization occur outside the body under conditions similar to those inside the body, followed by the transfer of zygote or early development stage into the female reproductive tract. Okay, so we want the correct similarity between ZIFT and IUT, right? Now, can you see third statement? It is written transfer of an embryo with more than 8 blastomere stage. It is in IUT but not in ZIFT. Artificial introduction of semen into the vagina, no. So, if we read the second statement, fertilization in both will occur outside the body, right? And there will be transfer of zygote or early development stage into the female reproductive tract. Okay, so in case of ZIFT, it is fallopian tube. In case of IUT, it is uterus. So correct answer should be 2. Next, statement 1 says the role of oxygen in the regulation of respiratory rhythm is quite insignificant, which is right. Every 100 ml of deoxygenated blood deliver approximately 4 ml of carbon dioxide to alveoli. This is also correct. So both the statements are correct. Correct answer should be 1. Yes. Now let's see regarding human excretion, which of the following is an active process? Okay. Glomerular filtration, see active process means which require energy or ATP. Reabsorption of water in PCT, reabsorption of urea from collecting duct, but reabsorption of amino acids. So active process will be reabsorption of amino acids and glucose as well. So correct answer will be 4, right? Next, animals that have had their DNA manipulated to possess and express a foreign gene can be used for study of diseases, testing vaccine safety, investigation of a new treatment for the disease, okay, and producing useful biological product. Okay, so all of them correct answer should be, that's why 3. Okay. Which of the following features is incorrect? with reference to a chemical synapse but correct with reference to electrical synapse. So what happens in electrical synapse? The presynaptic membrane is in close proximity with the postsynaptic membrane and there is direct flow of current or electricity or the action potential. Presence of gap junction, involvement of neurotransmitter, slower synaptic transmission, a wide synaptic cleft. So we want incorrect for a chemical synapse, right? Remember that slower synaptic transmission is there for chemical synapse, okay? And we want incorrect for chemical synapse, so this is true. Wide synaptic cleft is also true for a chemical synapse, right? Involvement of neurotransmitter is also true, but presence of gap junction will be incorrect for the chemical synapse and correct for the electrical synapse. So correct answer should be 1. Next. So we have assertion and reason here. During the post-industrialization period in England, the number of melanized moth increased in comparison to the white-winged moth. So what happened? At the time of industrialization, when the pollution increased, all the lichens that were growing in the bark of trees, they died, right? Apart from that, there was soot deposition on the bark of trees, and because of which the dark colored moths were not visible, but the light color moths were visible, okay, to the predators. So definitely there was increase in the number of melanized moth. Now after industrialization, the tree trunks became dark due to industrial smoke and soot, making the white-winged moth easily spotted and preyed upon by the predator, while the melanized moth survived. Absolutely. Okay, so assertion is true, reason is true, reason is even the correct explanation of assertion. So that's why correct answer will be 1. Okay. So with this we have discussed the questions of zoology. Now it is time to discuss the physics portion. So thank you very much students for attending the session.
पेन और ये चल रहा है ना, स्टार्ट करें. ये तो कुछ और चल रहा है इसमें. आगे है क्या? आगे तो नहीं है. Good evening to everyone. So let's start with a discussion of physics, right? Am I audible to everybody? Can anybody say, comment if I am audible? Let's start. Okay. So what is the first question? Just a minute. I think this. Okay. I think they have started from the back, so I'll just a minute. Okay. So let's start, right? Am I audible? Yes. Okay. Arpita, Vrishti, good evening. Right. So let's start. Okay. So this is the first question. So what it is saying, we have to match basically both the lists. So let us start. So whenever you are getting a matrix match, try to eliminate options and try to do it in a very fast manner. How you can do it? By basically eliminating options. So let us try to see how we can eliminate options. So if you see the options, we have 333 here, we have 111 here. So let us try to eliminate, start from the very simple one and try to eliminate options. So let us start with D here, right? Just a minute. Yeah, so let us try to do this. Okay, fine. So diatomic gas. अरे यार क्या हो रहा है? Yeah. So diatomic gas with one vibrational mode at constant volume. We have to match the molar specific heat capacity, right? So let us try to see. So what is the degree of freedom for a diatomic gas? Yes. So what is the degree of freedom for a diatomic gas which has one vibrational mode as well, right? So what do we have? Initially we have 3 as translational, 2 we have 3 is the translational, then we have 2, then we have the mode of vibrations also, right? So mode of vibrations, so how many vibrational degree of freedom it will have? 2 into number of modes. So number of modes are 1 here, so that is 2 more. So 3 plus 2, 5 plus 2, 7. So we have 7 degrees of freedom here, right? And the molar heat capacity at constant volume is nothing but CV. So CV would be, if you know, it is FR by 2, right? So that is 7R by 2, correct? So now we have, what do we have? For D option we have first. So I'll write first here, which means that if you see out of these options, these are anyway eliminated, right? So we'll have our answer from 1 and 2 only. So let us solve first, because that is the differentiator in both of them, right? So for solid, we all know what is the specific heat capacity. I mean, what is the molar heat capacity? That is 3R. This is a standard thing, right? Everybody knows this. So that is, for A we have 3. So now if we have 3 here and if we have 1 here, the answer would be 2, right? Fairly simple, correct? For carbon is an exception. For carbon it is actually 6.1 and that is less than R. So if you know, for carbon it is less than R. Again, water molecule is a triatomic molecule, so you can calculate by 3 into 3RT. So basically 3 into 3R is basically nothing but 9R, right? So for water molecule you have 4. Understood? So let's move ahead. Now a physical quantity Z depends on 4 observables A, B, C, D, right? And basically this is the expression that they have given you, and they have given you the percentage error in each of A, B, C and D, right? So what they have given you here, they have given you, so I'll write it here, they have given you delta A by A into 100. This is a percentage of error in A. They have given you that as how much? That is 2%, correct? They have given you error percentage in B also. That is nothing but 1.5%, correct? And so on, they have given you for C and D also, right? So now if I want to calculate the percentage error in Z, how do I calculate? Let me change the anyway, so let's do it with red pen only, right? So if I want to calculate the percentage error in Z, how do I calculate? So that is a standard that comes from taking log and then differentiating, right? So that is a standard, I'll just write it here. So this power 2 multiplied by delta A upon A, correct? Plus the power of B, which is 2 by 3 delta B by B, plus C to the power half. We have C to the power half, and you know, when you take it in the numerator it would be minus half, but since errors are always added into the system, errors are never subtracted, they are always added into the system. So what we will have 1 by 2 delta C by C, plus then we have 3 as the power, so 3 delta D by D, right? You multiply the whole equation by 100 and this is nothing but delta Z. This is basically the percentage error in Z, right? So if you multiply by 100 to the whole equation, what you will get? You will get 2 into percentage error in delta A. What is the percentage error in delta A? 2%. You write it 2 by 3 multiplied by 1.5 plus 1 by 2 multiplied by 4 plus 3 into 2.5, right? So now you calculate this. This is a normal calculation that you can already do, right? So your answer will come as 14.5%, correct? Let's move ahead. In a vernier caliper, one vernier scale division is always smaller than one main scale division. Okay. So they are saying that in vernier caliper, one vernier scale division is always smaller than one main scale division, and that is true, right? So if you remember, we write the least count of a vernier caliper as one main scale division minus one vernier scale division, right? So vernier scale division is lesser as compared to the main scale division, correct? So that is true, right? Now, what is the second statement saying? The vernier constant. What is vernier constant? Vernier constant is nothing but the least count only, right? So vernier constant is given by one main scale division multiplied by the number of vernier scale division. That is incorrect because it is the difference between the two, right? So that is false. So first statement is correct, second is incorrect. Understood? That is second. Now let's move ahead. Question number 4. A bird flies with a speed of 10 kilometers per hour and a car moves with a uniform speed of 8 kilometers per hour. Both starts moving from B towards A. Given AB is 40 kilometers at some instant, the bird having reached A flies back immediately to meet approaching car. Okay, as soon as it reaches the car, it flies back to A. The bird repeats this till both the car and the bird reach A simultaneously. The total distance flown by the bird is, right? So this is a fairly big question, right? And I've always told you, if you want to understand physics and if you want to do physics questions very, very easily, what you want to do is you want to visualize things, what is happening. So if you try to make a diagram here and then visualize, you will get a very clear approach of how this will happen, right? So let me draw a diagram for you. Rest you can do for yourself. So let's say, this is my point. So let us say that, there is a bird like this, right? This is a bird, correct? And we have a car also here. So they both are starting from here only, correct? So this is moving with 8 and this is moving with 10. And let's say this is A. Don't go on the figures. Don't go on the art. Art is not important. The concept is important, right? So now this is B basically. So this distance is given as how much? This distance is given as AB is equal to 40 kilometers. Just a minute. Anyways, this is 40 kilometers. Correct? This is 40 kilometers, right? Now, what is happening basically, let's say this, since the speed of this is more, you can see it is 10 kilometers, it will go here, return back, and while returning back, the car will move some forward distance, right? So it will meet here. As soon as it meets here, it again flies back here. It again returns back, car has moved somewhere here, it again goes like this, and it keeps on happening until the car and the bird both reach that place, right? A. So now if you see, it might sound difficult initially, but if you see what is one thing that both of them are following, that is they started from the same position and they are ending at the same position also, which is this wall A, correct? Now, for how much time has the bird flown? Can I calculate that? That is very, very easy. Why? How I can calculate that? Because I know that this is 8 kilometers per hour and this car is moving from B to A. And if I calculate that time of car moving from B to A, I will get the total time for which the bird has flown, right? So let us do that. So what is the time? Time would be 40 kilometers divided by the speed. What is the speed of this car? That is 8 kilometers per hour, right? So 8 kilometers per hour, which is giving me 5 hours. So basically for 5 hours the bird has flown, and I have to calculate how much total distance the bird has traveled. So that would be nothing but the speed of the bird, which is constant, into the time. So 5 into 10, which gives you 50 kilometers, right? Understood? Clear? So always try to draw diagrams first and then try to approach the question in those questions in which the diagrams can be drawn. So now let us see that. Now let us see the next question. A ball of mass 100 gram is projected with a velocity of 20 meter per second at 60 degrees. So let me draw a ball here. This is how much? This is your 20 meter per second, right? It is projected at an angle of 60 degrees, right? Okay. So basically it is a projectile. So what will happen? It will go like this and it will go like this. Correct? This is what is happening here. Yes? Now, we have to calculate the decrease in the kinetic energy. The decrease in the kinetic energy of the ball during the motion from point of projection to the highest point. So highest point is here, right? Now we can do the components of this 20. So we'll have one component here, another component here. Why I am telling you to do the component? Because this component never changes, right? And at the highest point, what is the velocity of this projectile? That would be the horizontal component of the velocity, which is nothing but 20 cos 60, right? So let us try to do this. 20 cos 60. What is cos 60? 1 by 2, right? The value of cos 60 is 1 by 2. So this will come out to be 10. Clear? Now we have velocity here. Can we calculate the difference in kinetic energies, right? Or the whatever they have asked, the decrease in the kinetic energy. You do half mv square here, you do half mv square here, then just take the difference. That is very, very simple calculation, right? So I'm not doing that because we have to cover a lot. Let's move ahead. A particle of mass 4m, which is initially at rest, explodes into 3 pieces of mass m and 2m. So what is happening? This 4m mass, so let's say a bomb is there, which is of mass 4m. Now this has exploded. This explodes. Now they are saying that it is exploding into 3 masses. What are those 3 masses? One is m, another is also m, and the third one is 2m, right? This is what the 3 masses are, m, and 2m, correct? Now the equal masses move along x and y axis with velocities 4 and 6. So these 2 masses, they are saying they are moving along the x and y axis, right? So let us try to move them. So this is moving here, this is moving here, right? And what is the velocity of these masses? 4 and 6. So this is 4 meter per second, this is 6 meter per second, right? This is in x axis, that is in y axis. Now you have to calculate the magnitude of the velocity of the heavier mass, which is the 2m, right? So how do we calculate this velocity of heavier mass? So what we do? We conserve the momentum. So in x direction the momentum is conserved, right? And in y direction also the momentum is conserved. This is a concept, right? So in explosion the momentum is conserved because there is no external force acting here, or the significant external force which is acting here, right? So let's say the velocity in the x direction is ux and in y direction is uy. We can calculate by momentum conservation. So ux into 2m ux into 2m, that has to be equal to 4 into m. So mm got cancelled, ux is nothing but 2. Ux is nothing but 2, correct? And in similar manner you can calculate y also. 2m into uy, that is equal to 6 into m. So uy is 3. Now you have both the components. Can you calculate the net velocity? What is the net velocity? Root over 3 square plus 2 square, correct? That is root over 13. Understood? Right? Fairly simple question. Nothing there to explain much. So in explosion questions, you have to understand that momentum is conserved along the x direction also, along the y direction also, and by momentum conservation you solve most of the questions. Is that very, very clear? Let's move ahead. Now, two spheres P and Q, each of mass 200 grams, so this is 200, this is 200, okay, are attached to a string of length 1 meter. So this is 1 meter of string which is attached here, okay, fine. The string and the spheres are then whirled in a horizontal circle about O. So both of this setup is basically moving like this, right? By connecting a string between O and P. So we are connecting a string between O and P and basically, whirling it around like this in a horizontal circle, right? At constant angular speed. So angular speed is constant here. The ratio of the tension in the string between P and Q to that between P and O is. So basically what are they asking? They are asking you the ratio of the tensions. Where are they asking you the ratio? They are asking you the ratio of tension here. Let's say the tension here is T1 and let's say the tension here is T2. So basically they are asking you T1 by T2, right? This is what they are asking you. Okay. So let us do it one by one, right? So we know that this whole setup is moving in a circular motion. Are yes or no? Is that clear? They are moving in a circular motion. So you can see that this Q, if I want to write an equation here at Q, this Q is facing if I sit on Q, there would be a centrifugal force that would be balanced by the tension, right? So this Q, can you tell me what is the centrifugal force which is acting here? That would be mr omega square, right? And what is m? M is given to you and both of the spheres have same mass. So I mean, I'm not taking this 200, I'm just writing m. M, what is r? R is from the center, which is 2. Mr omega square. This is my Q. Let's say the tension here is T1. So basically T1 is balancing 2m omega square. Yes? Similar equation we will write at P also, right? But at P you will see that there are two forces which are coming into play. In fact, three forces which are coming into play. So one is the centrifugal force which is coming outside, which is again m. R is 1 in this case. Omega square, this is 1. And you see this tension is also acting here. So this is T1 here which is acting here and there is this tension T2 which is acting opposite. So we will write again this equation. T2 is equal to T1 plus m omega square and we already know what is T1. T1 is 2m omega square. So you put 2m, you will get 3m omega square. That is T2. Now you take the ratio of T2 and T1, right? Is that clear? Sir, please change the ink. Which color do you want, beta? What should I put? I think this is better. Yeah? Let's move ahead. Okay. So you will get the ratio as 2 by 3, correct? Next. Sir, could you please change the color of the pen? Yeah, I have changed to yellow, right? Let's see. The coefficient of friction between the box A and the cart is 0.7, right? The box is of mass 2 kg. The minimum acceleration of cart so that block does not fall down is. So basically what is happening, this block is here, this is a cart and it is being moved with such an acceleration that this mass A is actually not sliding down, it is there only, right? So it is not attached to this cart but it is being held by force of friction, right? Why it is not falling down? There is a gravitational force which is acting. What is that gravitational force? M into G. But that is being balanced by the friction. That is why it is not, falling down. So let us make the FBT of this particle A. So what is happening here? To A, we have a normal reaction N, correct? Which is here, right? And since this whole setup is accelerating, I'll apply a pseudo force which is m into a, right? So we have the mass of this m into a, right? And there would be a friction which is acting, and what is that friction? Mu into n. And if you see, and if I balance it, n would be equal to m into a, and friction is nothing but mu into n. And what is n? N is nothing but m into a. Clear? And that is equal to m into g, right? So now can you balance? Friction would be equal to m into g. That is what we are doing, right? So now you do it. This is just the calculation part. So mu is given to us. We have to calculate what? We have to calculate the acceleration. So acceleration would be equal to g by mu in this case, right? Because mm will cancel out from both the sides. Yes? So 10 by, you can do it. So 9.8. What is the, let me just check once. So g by mu, what, can you calculate this g by mu? What is the answer coming? G, I think 9.8 divided by mu, what is mu? Mu is 0.7. Yes, so you can cancel it out. Yeah, so answer will come, that is 14, right? Clear? That is 14. Let's go, let's move ahead. A sphere of mass 2m collides with a stationary sphere of 3m. So we have a 2m mass which is moving with some velocity. It is colliding with a stationary mass. So this mass is stationary and when it collides, it sticks to it, which means that now both are moving together, right? Let's say they are moving with some velocity v2, right? Rishi Chandra 14, yeah, that is correct. So let us do this. So again, we will do the momentum conservation, right? So let us do the momentum conservation. So what is the initial momentum? 2m into v, that is equal to the final momentum, which is 5m into v2. So mm cancels out. What is v2? v2 will come out to be 2v divided by 5, correct? That is my velocity. That is my final velocity, correct? Okay, now what they are asking, the loss in the kinetic energy is very, very simple. You can calculate the final kinetic energy, that is equal to half. What is the total mass, which is 5m, into velocity, final velocity. What is final velocity? 2v by 5 whole square. This is my final kinetic energy. What is my initial kinetic energy? That would be half 2m into v square, right? Now, how to calculate the percentage? Final minus initial upon initial into 100. So that is very, very simple. K2 minus K1 upon K1 into 100. That I am leaving up to you because I know you are very talented in maths, right? So that you can do by yourself, correct? So the answer will come as 60%, that you can do, right? Let's move ahead. A uniform cable of mass m and length L is placed on a horizontal surface, right, such that its 1 by nth part is hanging below. So let's say this is the table, right? So this is the table, let's say. Now we have a hanging chain like this, right? Let's say this is something which is hanging here, right? Correct? So now they are saying that 1 nth of the part is hanging. So if this is a total length, some part which would be L by n, that would be hanging down, right? So this part length is L by n, correct? What would be the mass? Again the same, that would be m by n, right? If total mass is m, this mass would be m by n. Now we have to basically pick it up and put it on the table, right? And now we are being asked what is the work done, right? So there is a very simple thing to do. Although the actual derivation comes by integrating, taking a part and then integrating, but then we are not going into that. What we will do is we will take the center of mass of this part, right, which is at the half distance, and that would be L by 2n. And when can this part come above? When the center of mass comes above, right? So what I'll do, I'll pick this up until the center of mass comes to the table. I will not leave it, right? So basically what I'm doing is I'm carrying this. I can assume that this whole part is at the center of mass. So this is the mass m by n at a distance of L by n from here, and I have to pick this mass and take it through a distance of L by n, and that would be nothing but the work done would be force, which is mg by n. This is L by 2n actually, L by 2n, because this center of mass is at a distance of L by 2n. So you will get the answer as mgl by 2n square. Correct? Clear? Let's move to the next question. The relation between T and displacement of a particle. This is a good question actually. Relatively better question. The relation between T and displacement x of a particle of mass m kg is given by this relation. So they have given you the relation between time and the displacement, right? Which is T is equal to root x plus 3. So from here I can write this also. T minus 3 whole square is equal to x. I can write this. You can take this 3 here and then square both sides and get a relation between x and t. Why I am writing this relation? Because I want to calculate the velocity first. Whenever such equations are given, you have to understand what is happening in the motion, right? For that you need different parameters like displacement, velocity, acceleration, right? So if I want velocity from here, what will I do? I'll differentiate this. That will give me the velocity. And what will this come out to be? 2 times of t minus 3. I hope all of you know this basic. 2 into t minus 3, that is my velocity. And if I differentiate this again, what will I get? I will get acceleration, right? So dv by dt is acceleration, and that is nothing but 2. So you can see that acceleration is coming constant, but velocity is not coming constant. Velocity is a function of time, and the displacement obviously is a function of time, right? So let us see what is happening here. If I see the motion at t is equal to 0, because I have to find the total work done in 6 seconds, so I need to know how is the motion happening, because I need displacement for that, right? And I need mass for that. I need displacement and force for that. So let us see what is the displacement. So at t is equal to 0, if you see what is happening, at t is equal to 0, what is the velocity? Velocity is minus 6. Let's say this direction is positive, this is negative, which means that the particle, wherever is the particle, it is moving in the negative direction with a speed of 6 meters per second. Correct? Understood? Right? Now, at t is equal to 3, if I ask you what is happening at t is equal to 3, if you see at t is equal to 3, if you put t is equal to 3, velocity will become 0, right? Which means that from t is equal to 0 till t is equal to 3, this particle has decelerated, right? And I can say that why? Because you see acceleration is 2, which means that acceleration is in this side, velocity was opposite, which means that this particle was going here decelerating, and finally the velocity became 0 here, right? In first 3 seconds, how much time it took to make it 0? 3 seconds. Now what will happen? Again, the acceleration is here only. So what will happen? It will accelerate. For another 3 seconds it will accelerate and come here again with the same velocity, which is 6 meters per second. You don't even need to calculate it. If you have done many questions, you know that this is going to happen, right? This particle is decelerating first for first 3 seconds and then accelerating for the next 3 seconds, which means that this displacement, if you see from here till here, then here, it is coming back to the same point, which means that the displacement is 0. And if displacement is 0, the work done has to be 0. There is no need to calculate work done here, right? Because that would be 0. Understood? Is this very, very clear, right? Can we move ahead? Let's move to the next question, right? A force F A I cap plus 2j cap is acting at a point. So let's draw a point. So this is a point. Let's say the force is acting in this direction. So it has two components, AI cap. So we have AI cap and we have 2j cap. This is my force. Is acting at a point whose position vector is given by 2i cap plus bj cap. 2i cap plus bj cap, correct? So this position vector is how much? This position vector is 2i cap. So 2i cap and 2j cap. Now they are saying that the angular momentum about the origin to be conserved. When is the angular momentum conserved? When the net torque acting on that point is 0, right? This is the concept. What is the concept? The net torque has to be 0, then only we can conserve the angular momentum. And what is net torque? Sir, how can we say that? So Madhvi is asking, sir, how can we say that it is accelerating for the next 3 seconds? Because Madhvi, we are saying that why is it accelerating? Because you see the acceleration is in this direction, correct? And it is starting from rest because here the velocity has become 0, right? but acceleration, the force is acting in this direction only, which means that the velocity will keep on going to increase now. Yes or no? Understood? When acceleration and velocity are in opposite direction, it decelerates. When acceleration and velocity are in same direction, it accelerates. Yes or no? I hope this is clear, right? Now, coming back to this question. So, net torque is 0, I know. And how do I write the net torque? Net torque is nothing but R cross F, where R is the position vector and F is the force vector, right? When can this R cross F be 0? When the angle theta between them is 0, which means that R and F, both of these vectors, they have to be in the same direction, or at least they should be parallel, right? And when can both of these vectors be parallel? When their slope is same, then only they can be parallel, right? So I can directly say what is the slope of this F vector? That is 2 by A, and that has to be equal to the slope of this vector. What is this vector? B by 2, which means that 4 is equal to AB, right? Now you will say that, sir, this is just one equation, how we will calculate? You can check for the options. Let us see where is AB coming as 4. In first option, is it coming? No, it is not coming, that is 5. In second option, it is coming. No, it is not coming, that is 2. Again, third option is 2. Only option is 4. Understood? So this is 4, correct? Is this very, very clear? Right? Let's move ahead. So you have to try to do questions fast by trying to eliminate things, right? And trying to do it, don't go for bigger methods, right? You don't have to solve each and every step. Try to do it in a very, very short way, right? Understood? Let us go on to the next question. So I'll move fast now because the time is going. Read each statements below carefully and select the true statements of the following. During rolling, the force of friction acts in the same direction as the direction of the motion of the center of mass. Not necessarily, right? It is not necessary that it is acting, because let's say this, if it is rolling in the if it is doing pure rolling in this on the fixed surface, the friction might act behind also, right? Not pure rolling, let's say this is the velocity and this is omega, right? It is not necessary, by the way, right? So if let's say V is not equal to omega r, then friction acts, right? If V is not equal to omega r, then friction will act, and friction will act in such a way that it tries to make V equal to omega into r, right? So I'll give you an example. Let's say in this case, V is greater than omega r. Just for an example, let's say this V is greater than omega into r. Then what will happen? If you see this point, if you see this A point, at this A point, V is here, omega r is behind, and V is not equal to omega r, and I am saying that V is greater than omega r. Now, where will the friction act? Friction will act in such a way that to make this V equal to omega r. And how it can make this V equal to omega r? By decreasing the velocity. And for decreasing the velocity, the friction has to act in the opposite direction, right? So we can see that it is not necessarily in the direction of the motion of the center of mass, right? So that is incorrect. Clear? Next, the instantaneous speed of the point of the contact during pure rolling on a fixed surface is 0. That is very, very true. If pure rolling is happening, what happens in the pure rolling? This relative displacement of this point is not happening, right? So the relative displacement of this point is 0. It can happen only when, if it is rolling on the fixed surface, it can happen only when V is equal to omega r, right? So it is asking the instantaneous speed. Since the relative displacement is not happening, what will happen? The rate of change of displacement is 0, relative displacement is 0, which means that it is 0. So B is correct, right? The instantaneous acceleration of the point of contact during rolling is 0. Not necessary. Not necessary that it is 0, right? It is written rolling, not pure rolling, right? It is rolling. Again, for D, for perfect rolling motion, or perfect rolling motion is nothing but pure rolling, the work done against friction is 0. That is correct, right? That is correct, because in pure rolling, what is happening? That point is not being displayed. That point is not being displaced. And if this point is not being displaced, the work done would always be 0 because the displacement is 0, right? Understood? Next, a wheel moving down a perfectly frictionless inclined plane will undergo slipping motion. perfectly frictionless surface. Yes, that is correct, because the friction is not there, which means that the slipping will happen now, rolling will not happen. So that is also correct. So your answer would be B, D and E. Is that very, very true? Yeah. So the answer is B, D and E. Is that very, very clear? Right? Anime, hello, you are late to the party. We have already started. So let's do the next question. Okay. During thermodynamic process, which of the following is path independent? Sorry, pardon. Which of the following is path independent? Right. So this is very fairly simple question. I don't need to even explain it. Whatever is the state function, that is path independent. So internal energy is basically state. It depends on the state. It does not depend on the path. Right. So that is the third option. Other heat absorbed and both of this work done, they both are path dependent. They depend on the work, right? When potential energy of a particle executing SHM is 1/4 of its maximum value, right? So let's say what is the concept here again? So let's say the amplitude of this SHM is A. So what is the maximum potential energy? It would be at the extreme position. And how much is that? Half K A square. That is my maximum potential energy. So it is saying that when potential energy of a particle executing SHM is 1/4 of its maximum value. So what is generally the potential energy in an SHM? 1 by kx square, right? So now they are saying that this 1 by 2 kx square, that is nothing but 1/4 of half ka square, right? So this half k gets cancelled from both of the sides, and what will x come out to be? X will come out to be a by 2. Fairly simple question, right? That is a by 2, right? That is very, very clear. Let's move ahead. The escape velocity from a spherical planet is VE. What is the escape velocity corresponding to another planet of twice the radius and half the mean density? So let us write the relation, right? So let's say that planet radius is RP. So that RP is what they are saying, it is twice of the radius, another planet of twice the radius. So RP is twice of RE, and the density they are saying is half. So density of this planet is half density of the previous one, right? Clear? What is the escape velocity? You know that escape velocity is directly proportional to mass by radius, because it is root of 2 gm by r, if you remember the formula, right? So it is directly dependent on mass and radius. So if I calculate the relation between mass and radius, I will get the velocity. So for mass you have to use this. So what is density? Density is mass per unit volume. So mass of that planet divided by volume of that planet, 4/3 pi radius cube is equal to half mass of that mass of the other planet, 4/3 pi RE cube, correct? 4/3 pi gets cancelled, right? So you can get the ratio here, mass MP by ME, that is equal to 1/2 RP by RE whole cube, correct? This is the relation, right? RP by RE I already know. What is RP by RE? That is 2, right? So you can write it here, 1/2 into 2 cube, which is 8, that is equal to 4, right? Now this you already know. Now you take the ratios for both of them. So VP is equal to root over, directly proportional to mass of planet upon radius of planet, and that is this. Now you take this ratio, VP by VE. You will get what? You will get root over MP by ME multiplied by RE by RP. Now you replace this, all these numbers here, you will get the answer. Is that very, very clear, right? Next. The gravitational potential energy due to the earth of a rocket at a distance d from the earth is minus u. Okay, so what is the gravitational potential energy? Minus g capital M small m by distance between them. So at a distance d, that is equal to minus u. So from here you can calculate u. What is u? U is g capital M small m by d. The weight of the rocket at that distance is, what is the weight of the rocket? Weight of the rocket is nothing but the force which is exerted, right? So what is the force exerted? G capital M small m by d square. or I can say U by D directly, because U is nothing but this. Clear? Is that very clear? Any doubts? Let us move on to the next question, right? Okay. Two bodies are having kinetic energies in the ratio 16:9, right? So what do we have? We have kinetic energy of 1 divided by kinetic energy of 2, that is 16/9. What are they asking? If they have same linear momentum, so linear momentum for both of them is same, then the ratio of their masses is. So basically you have to depict kinetic energy in terms of momentum. So what is kinetic energy in terms of momentum? P square by 2m. Yes. Now can you put it? Since P is same, you will get m2 by m1 as 16/9. But what are they asking? They are asking the ratio of masses, which is m1 by m2. That would be 9 by 16. Clear? Is that very clear? Let's move ahead. Okay. Consider the following two statements 1 and 2 and identify the correct choice. The excess pressure inside a small liquid drop is more than that of a big drop. That is correct, because excess pressure, if you know, is inversely proportional to the radius, right? If it is a drop, it is 2T by R. If it is a soap, that is 4T by R, right? So it is inversely proportional to the radius. The smaller the radius, greater would be the excess pressure, right? So yes, that is true. I mean, for small drop, it is more than big drop. So that is true. So this is true. What is the second statement saying? A liquid wets a solid if the angle of contact between them is acute. That is again correct, right? So that is fairly simple question. Let's move on to the next question. Two soap bubbles of radius 3 and 6 respectively are in contact with each other. The radius of curvature of the common surface is, okay. So again, so what is happening? One is smaller, another one is bigger. So they come in contact. What will happen? So basically, you know, there would be this kind of surface. So I'll try to make this surface. It will be something this kind of surface. This is 3 centimeter. This is 6 centimeter. Right? So again, the radius of the surface of the common surfaces. So very simple. Can you write it for the the smaller soap bubble? P1 minus P0. What is the excess pressure? 4T by R, which is 3 centimeters. For second, you can write P2 minus P0 is equal to 4T by 6. Right? So here you have pressure P1, here you have pressure P2. We need to find P1 minus P2. I can write P1 minus P2 as 4T upon the radius of this bulge which is there, right? So I'll subtract both of them. So if I subtract both of them, I'll get P1 minus P2 here, which I can write 4T by R equivalent. That is equal to 4T times 1 by 3 minus 1 by 6. So this cancels out 1 by R equivalent. From here you can calculate, right? So you'll get the answer as 6 centimeter. 1 by 3 minus 1 by 6 is 1 by 6 and R equivalent would be 6. Clear? For a given mass of gas at two different constant pressure P1 and P2, graphs are drawn with absolute temperatures. So you can see that this scale at Y is nothing but V by T. So you can see that V by T for 1 is more than V by T for 2. So V by T for 1 is greater than V by T for 2. And what is V by T? So if you remember, PV is equal to nRT, and from here you can write V by T as nR by P, right? So basically what they are saying is that 1 by P1 is greater than 1 by P2. This is what they are saying, yes? And from here you have to calculate the relation. Very simple. P2 is greater than P1, right? If you reciprocal, what will happen? The pressures will, this relation will reverse, right? P2 will be greater than P1. Two rods of same area of cross section but made of different materials and having lengths L and 2L. So we have one rod whose length is L, another rod whose length is 2L, okay fine, are welded together. So they are welded together and total length will be 3L, okay. If 2A and A are the linear coefficients of expansion, so alpha for this is 2A, alpha for this is A. Then the linear coefficient of the expansion of the composite rod would be? Very, very simple. So if I take this composite rod of length 3L, let's say the linear coefficient of expansion is alpha. So if I expand it by the same temperature, what will happen? Delta L will be equal to 3L alpha delta T. Yes? And in this case, what will happen? There would be a change in first length, there would be change in the second length. Right? There would be a change in first length, that would be L alpha, which is 2A delta T. In second, what would be the change? 2L alpha delta T. And I know that equivalent would be nothing but L1 plus L2. Right? So you have these two equations. Now you put it and you will get the answer. Very, very simple. Right? Clear? Okay. Next question. Hot coffee is found to cool from 90 to 80 degrees Celsius in 2 minutes. Okay. So we know that basically, the rate of change of temperature is directly proportional to the difference in the temperature, T minus Ts, where Ts is the temperature of surrounding and T is nothing but T1 plus T2 by 2. Correct? So this you have to use here. Correct? So from 90 to 80. So from 90 to 80, it is going in 2 minutes. So let us apply that. What is dt? dt is 10. So 10 by 2 minutes, which is change in time, that is directly proportional to capital T. What is capital T? T1 plus T2 by 2. That is 90 plus 80 by 2, which is 170 by 2 minus surrounding temperature. What is the surrounding temperature? That is 30. So this is first equation. The time taken for the same coffee to cool from 80 to 70 is, again for the second case you have 80 to 70 and you have to calculate the time. So 80 to 70, again the change is 10, time let's say is T, is directly proportional to 80 plus 70, that is 150 by 2 minus 30. Now you take the ratio of both of them, you will get the time, right? Clear? Simple. The equation of a transfer wave traveling along a string is given by 4 sine blah blah blah, but this is in mm, where x is in mm and t is in second, the velocity of wave is. So this is very, very simple, right? So k we already know, 20 into 10 to the power minus 3, omega we know, what is omega? 600, and we know the relation, k is equal to omega by v. So what is v from here? Omega by k. So 600 divided by 20 into 10 to the power minus 3, but please remember this is in mm per second, right? Not in meter per second. But the answers are in meter per second, right? So you have to convert it. So that is how much? Can you do it now? 3 into 10 to the power 4 mm per second. This is mm per second, right? So in meters it would be multiplied by 10 to the power minus 3, right? Clear? So that would be 30 in magnitude. But let us also see the sign also, right? So this is a speed, this is not the velocity, because velocity comes with sign. And how do I see in which direction is the wave moving? This and this both are positive, which means that the wave is moving in the negative direction, right? So if it is moving in negative direction, this would be, the velocity would be actually minus 30, right? This is the speed. Correct? Understood? Next, can we go for next? 25th, okay. A concave mirror of focal length f in air is dipped in the liquid of refractive index mu, its focal length will be, right? Okay. Just a minute, let me see which question is this. I have the paper with me. So actually this answer is wrong, I think. This is not 2. This is wrong. Because concave mirror, so whenever you dip a mirror into any refractive index, the focal length doesn't change, right? So that is wrong. The answer is 4, F. So it will not change basically, right? I think this is fairly simple. This is wrong. Okay, next question. The refractive index of the material of a glass prism is √3. The angle of minimum deviation, okay, So this is a prism, minimum deviation, the light becomes parallel and it goes like this, right? So the refractive index is √3, okay. The angle of minimum deviation is equal to the angle of prism. So angle of minimum deviation, delta min, is given by I plus E minus A, and at minimum deviation I becomes equal to E, so that is 2I minus A. Now they are saying that this minimum deviation is nothing but the angle of the prism, which means that this is A. From here you can see that I would be equal to A, right? Understood? Now at minimum deviation I know this R1 and R2, they both are equal and they both are equal to A by 2, right? So now I can apply the Snell's law here. You apply the Snell's law here, right? What is the Snell's law? Sine of I is equal to √3 times sine of basically R1, which is nothing but A by 2. So that is A by 2. And what is sine I? Sine I is nothing but A sine A. So sine A is equal to √3 times of sine of A by 2. I can write this sine A by 2 as 2 sine A by 2 cos A by 2. That is equal to √3 sine of A by 2. This sine A by 2, this sine A by 2 gets cancelled and you will get cos of A by 2 as √3 by 2, which means that A by 2 is nothing but, how much is A by 2? So cos 30 is basically √3 by 2, which is, this is equal to pi by 6, and that is equal to 3, right? So you get A as 60 degrees. Is that very clear, right? Next. Given below are two statements, one is labeled as A and other is labeled as R, okay. In Young's double slit experiment, the fringes produced by red light are closer as compared to the produced by the blue light. So when fringes, if they are asking you the closeness of the fringes, what they are asking you? They are asking you the fringe width. What is the fringe width given by? Fringe width is given by d lambda by small d, right? D lambda by small d, and lambda is nothing but the wavelength, right? So if lambda is the wavelength, lambda of, you know, that red light is more than lambda of blue light, correct? So if lambda of red light is greater than lambda of blue light, the fringe width of red would be greater than fringe width of basically B, right? Understood? So what are they saying? The fringe produced by red light are closer, that they are not closer, right? Just a minute. So they are not closer, they are basically farther away. So this is wrong, right? The fringe width is directly proportional to the wavelength of light, that is correct. Clear? Right? That is clear. Let's move to the next. As shown in the diagram, two fixed charges Q1 and Q2 are there, which are 0.2 meter apart, whereas the net field 0. So this charge is 1 and this charge is -4. So basically that point would be somewhere here. So let's say that distance is x. Now you write the net field here. What is the net field due to this charge? KQ, which is 1 by r square, which is x square. What is the field due to this -4? That would be K4. I've already shown the direction, so I'm not writing -4 by x plus 0.2 whole square. Right? So they both should be equal. So you can see that from here x plus 0.2 whole square divided by x square, that is equal to 4, and from here you can calculate the value of x, right? That is clear. So it will come out to be 0.2, right? Which of the following can intersect each other? Electrostatic field lines, obviously not. They cannot intersect with each other, right? Streamlines in turbulent flow, yes. In turbulent flow there is no structured flow, right? So they can intersect each other, but in laminar flow they cannot, right? So obviously in this they cannot. Equipotential surfaces, again they cannot, right? Clear? All of the condensers in the circuit shown are equal and each is 5 millifarad, okay? The equivalent capacity of the terminals is. So you can see that this whole part is useless. Why it is useless? Because the starting and end are the same. So this is short circuited. So you can remove this. This is equivalent to a simple circuit like this. If this is C, this is C, and they both are in series, you will get C by 2. And what is C? C is nothing but 5 millifarad. So your answer would be 2.5 millifarad, right? Understood? Clear? Is this clear? Next. What is the potential difference VA minus VB in the circuit? So again, very simple. You start from here, we end here. We have done this in previous discussion also. So you start from here, VB. Now you have to note that this is the resistance here, right? So there would be a potential drop by how much? I into R, correct? And then this is a gain in potential, so that would be plus 6. Again there is a resistor, there would be a potential drop by how much? Minus 2 into 2, that is equal to VA, correct? So from here you can calculate. What is this coming? VA minus VB is equal to 6 minus 8, which is minus 2. 6 minus 8, which is minus 2. Madhvi is asking, sir, how do we do equivalent capacitance type problems easily? So Madhvi, the capacitance problems are same as resistance problem. If you know how to do resistance, the same has to be replicated here. Same, exactly same. So when they are teaching you current, electricity, and they are teaching you how to calculate equivalent resistance, you can practically replace all these resistances and keep a capacitance and solve similarly, just that the formulas are different, right? So for the resistance, the series is R1 plus R2. For capacitance, it is 1 by C is equal to 1 by C1 plus 1 by C2, but the concepts are same. Clear? The series and parallel concepts are same, right? So again, you have to go back, revise your notes. The Wheatstone bridge, which is applicable in resistance, it is applicable in capacitance also. Clear? Sir briefly explains both. I cannot, because the time is not there. We have to do a lot of questions. So yeah, maybe you can ask your lecturer who is delivering you who is teaching you physics. They can explain you better. I mean, this is not the place to explain. This is not the class. This is the paper discussion, right? Okay. So resistance in two gaps of a meter bridge are 10 ohm and 30 ohm. Okay. So let's say we have a resistance here. This is 10 ohm. Another is 30 ohm, and we have this meter bridge like this here. So let's say this length is x. Total length is 100 centimeters. So that would be 100 minus x, right? So now they are saying that this is a meter bridge situation. So again we know that x upon 100 minus x, that is equal to 10 by 30. So from here you calculate x, right? You will calculate x. So let us try to calculate. So from here x will come out to be 25. Now they are saying that you interchange both of them. Now you make this 30, you make this 10. Again what will happen? Just the equations will change. X upon 100 minus x would this time be equal to 30 by 10. So from here you will get x as 75. And they are asking you the change. What is the change? 75 minus 25, that is 50, right? 75 minus 25, that is 50. Clear? Next. Which of the following resistivity versus temperature curves is most suitable to be used in a wire bounded state? So this is again basically a little change comes, right? So resistivity basically varies with temperature, generally linearly. Like this only it varies, right? But very little, right? NEET is this paper easy, moderate, tough, and scoring 100 in this paper is safe. Okay. With respect, yes, with respect to NEET, this paper is moderate. Some are good questions, right? So I think again it depends what are you targeting. If if you are targeting yourself into the top 1 percentile, then obviously you need to score more than 100, right? Let's move ahead. An electron is projected with uniform velocity along the axis of a current carrying long solenoid. Which of the following is true? Right? So we have a long solenoid here. So you remember that for long solenoid, the magnetic field inside is constant, and that is given by mu naught Ni, if you remember, right? So that is a uniform magnetic field which we have. So we are projecting an electron here, right? So like this we are projecting an electron with uniform velocity, and since the electron is projected in a similar direction as compared to B, the force which will be acting that would be 0 because V cross B would be 0. V and B are parallel, so this would be 0, which means that there is no deflection here, right? The electron will continue moving in the same path with the same uniform velocity, right? So that won't affect, right? Again, let us move ahead. A circular coil of radius r is carrying current I. Okay, fine. So we have a circular coil which is carrying of radius r, which is carrying current I. The ratio of the magnetic field at the center of the coil. What is the magnetic field at the center of the coil? That is mu naught I by 2 r, right? And at a point at a distance root 8 r from the center of the coil on the axis. So this is the axis. So let's say this is a point and this distance is root 8 r. So there is a standard formula. I think most of you would know. Mu naught I. What is the formula? Can anybody tell me? Mu naught I by 2 r square upon r square plus x square to the power 3 by 2. That is the formula of magnetic field on the axis of a coil, right? So you put that root 8 r here. So you will get on the axis mu naught I by 2 r square divided by, so root 8 will become 8. So 8 plus 1 will be 9. 9 is nothing but 3 square, and then there will be 3 cube. So that would be 27 r cube, and that would be equal to mu naught I by 54 r. Now you have to take the ratio, right? So that would be 27:1. Easy, right? So this is a formula based question. So for NEET, please remember that it's okay. So these test papers are for making you know your weak areas. So even if you lost in some parts, try to improve next time, right? The inductance of a coil in which a current of 0.1 ampere increasing at the rate of 0.5 represents a power flow of 0.5 watt. So what is the inductance? So very simple. Power is nothing but V into I, and what is V for inductance? That is L di by dt, right? So power would be nothing but L di by dt multiplied by I, correct? So power is given as 0.5, that is equal to L. What is di by dt given? 0.5, so that is 1 by 2 multiplied by current, that is again 0.1. So you get L as 10, right? You get L as 10. Is that very clear? Next. The ratio of secondary to the primary turns in an ideal transformer is 5:4. If the power output be P, then the input power. So again, if it is an ideal transformer, right? In ideal transformer, the input and output are equal. Power input is equal to power output. It does not depend on the ratio, right? So that is equal to P, correct? Clear? In an AC circuit, the instantaneous values of EMF and current are this. So 200 sine 314 and I is given. What is the average power? So what is the formula for average power? Average power is nothing but V RMS I RMS cos phi, where phi is here. You can see that what is the phase difference between both of them? That is equal to pi by 3, correct? And what is VRMS? VRMS is V naught by root 2. IRMS is I naught by root 2 cos of pi by 3. Can you put these values, please? What is V naught? V naught is 200. What is I naught? I naught is what is I naught? I naught is 1. So 1 divided by 2 cos of 60, which is 1 by 2, right? So that would be 50, right? So that would be 50. Is that very clear, right? Again, let us see this question. In this question also you can use the technique of elimination, right? Okay. So you have an LCR circuit. So you have an LCR circuit connected to an AC source, right? Which one of the graph represents the variation of current I with frequency? Let us see how to eliminate question. Sir, if one is in sine, other than cos, please. No, so basically what you are saying is, if both of them are in different, one is in sine, one is in cos, you basically convert both of them into sine, right? Understood? By adding or subtracting pi by 2, right? You have to see what is the equation such that it doesn't change, correct? Clear? Next, let's see this. So now they are asking you what is the function of current with respect to frequency. Let us try to eliminate this. So if I want to calculate I naught would be nothing but V naught upon Z. And what is Z? Z is nothing but the impedance, right, of the LCR circuit. V naught by Z. Okay, fine. Okay, fine. What is Z? How do I write Z? So Z is basically, where do I write? Z is basically, if you know, capital R square plus XL minus XC whole square. What is XL? XL, if I want to write it in terms of frequency, that would be 2 pi f into L, and XC would be nothing but 1 upon 2 pi f into C, right? So that is XL and XC with respect to the, yes, that is correct. XC 1 by 2 pi FC, right? Now, you see that, now my I is dependent on Z. When is Z, minimum? Z is minimum at, if you remember, Z is minimum when XL would be equal to XC, and that is called the resonance, right? At this it is 1 by root over LC, you get omega as 1 by root over LC, and at this point basically XL is equal to XC and Z is minimum. When Z would be minimum, I would be maximum, right? So that will come at one point of time in the graph, right? So if XL is equal to XC, for that value of F, I will get I as maximum, right? In all these four options, if you see, where can you see the maximum, such a graph where you are getting a maximum current? Here you are getting the minimum current, so this will not be the option. Here again there is no such thing as maximum, right? Here also there is a minima. Only in this question there is a maximum. I mean, this option there is a maxima varying with that, right? So obviously this is not a constant function. You can see that this is not a constant function, right? So that you will get as 3. Is that very, very clear, right? So this is how you can eliminate this without even solving. A plane electromagnetic wave of frequency 50 megahertz travels in free space along the positive x direction. E is given. Again, very, very simple. You have to calculate B. So E is equal to C into B. This you have to do. I am not doing it. Very, very simple. So let's move ahead. The work function of cesium and lithium are this and this. If we incident a light of wavelength 450 on these two metals, then the photoelectric effect is possible. So when is the photoelectric effect possible? Botany ma'am got 100 centimeter. Anyways, just a minute. So if we incident a light of wavelength 450 nanometer on these two metal surface, so basically when will the photoelectric effect happen? When basically the energy that you are giving, that is more than the threshold, right? So how do we calculate the incident energy? That is SC by lambda, right? So that is nothing but 1240 divided by 450 in electron volts. You calculate this value and you compare this value with 1.9 and 2.5. I think this was coming out to be 2.75 or something. Please check it once again. This is greater than both of these, and which means that in both of them the photoelectric effect will happen, right? Clear? Given below are two statements. One is labeled as assertion, other is labeled as reason. The binding energy per nucleon is found to be practically almost independent of the mass number A for nuclei between 30 and 170. That is correct. If you remember the graph, of binding energy per nucleon, it is something like this, right? So in this range it is almost constant, right? Clear? Nuclear force is a long range force. Obviously this is wrong. It is a short range force, right? So R is false. In the given circuit, the current flow through the PN junction would be. So again you can see that this is at the higher potential, this is at the lower potential. So this is higher, this is lower, which means that this is a forward bias diode, right? Which means that since this is ideal, it will act as a wire, and if it acts as a wire, this is short circuited and we don't need this, which means that the resistance in the circuit is only 5 ohms, right? And what would be the current? Current would be 10 by 5, which is 2 amperes. Clear? Is that very, very clear? Next, the logic circuit. Who was asking the logic circuit? The logic circuit shown below has input wave A and B as shown. Pick out the correct output form. Yes, that is correct. Nuclear force is a short range force. It acts between the nucleons, right? So now again, this is very, very simple. Just a minute. So this is the output that is given to you, and we have the input. So in such questions, what do? You write the values here. If you give 1 here and give 1 here, what will happen? If you give 1 1, you will get 0 here, 0 here. This is a NOT gate. This 0 0 will plus, and then you have again a NOT, so that will again you will get 1. So when you have a high and when you have a high, you will get a high. Where is high? So out of these 3, I will have 1 as the answer, right? So let's see. Again, so you are giving 1 1. So this is what? This corresponds to 0. This is 0. This is 1. This is 1. So if I give both of them 0, what will happen? If I give both of them with 0, this will become 1. This will become 1. 1 plus 1 is 1, and this will give me 0, right? So for this second part, you will get 0. So 0 I am getting in all of them actually. Okay, all of them I am getting 0. So like this you have to do, basically you have to see what is here and what is there. So again it is 0 here, this is 1 here. So you have 0 here, you have 1 here. So this will become 1 here and this will become 0 here. So again this will become 0 here, right? So again for the third part also you will get 0. So where is this 0? Here you have 0, here you have 0, here you don't have. So this is gone, this was already gone. So like this you have to basically do it, right? Clear? This is very, very simple. You can do this. Yes, by using Boolean algebra also you can do it, right? But if you forget that Boolean algebra formula, then you will get the wrong answer. Yes? Clear? So when you are in doubt, please start putting the values and do it. It will give you a quick answer. So this is the last question. A sinusoidal voltage of peak value 200 volt is connected to a diode and the resistor R is shown. Wave rectification occurs if the forward resistance of the diode is negligible compared to the R, then the RMS voltage is. So basically, so this is a half wave rectification, which means that half of it would be transferred to the resistor. So very, very simple. Out of that 200, 100 would be transferred, right? That is clear. So with this officially we are done, right? We are done with this discussion. I hope it is useful to you all, and please focus on the concepts rather than the cramming part, right? Thank you so much.
Hello students, today's NEET grand test paper chemistry discussion we'll see. Let us see the first question. N plus L value for the valency electron of sodium is X. In the case of what you can say sodium Na, you have 3s1. And what will be the N value in this case? N value is 3 and the L value for S is what you can say 0. So therefore it is 3 here. Continuation in the question, for the differentiating electron of scandium is Y. N plus L value for the scandium he is asking. So scandium in this case we'll write, this is 3d1 is a differentiating electron for the scandium. So therefore this will be what you can say 3 and L value for the D will be 2. So therefore this will be 5 and the sum of X and Y value he is asking. So in this case it becomes 8. So the first option, the first question The correct answer is 8. Okay, that's what you have. Now let us move to the second question. Non-directional bond. You know, non-directional bond is always generally used with reference to ionic bond, because what happens in ionic bond, you have what you can say component like Na plus is surrounded by 6Cl minus. So with which one it has actually formed the bond, we cannot say. So every ionic bond, let it be NaCl, cesium chloride, or in this particular case, first, second and fourth are covalent compounds, whereas Kf is ionic compound. So ionic bond is considered to be a non-directional bond. Among what you can say options, there is only one ionic compound, so that is Kf. Now let us see the next question. Incorrect trend is, here nickel, copper and zinc, they have given atomic radius. If you just see 3D series we have scandium, titanium, vanadium, chromium, manganese, iron, cobalt, nickel. We have nickel here discussion, copper and zinc. Here what happens in the atomic size, scandium is having larger size. Initially there is a decrease in size, then it remains what you can say same, then slightly what you can say increases at the end. So here they have given nickel is smaller than copper and zinc. That's right, because of completely filled D, it is having repulsions and slightly size increases, but still zinc will be having smaller size when compared to the scandium. So the first option, nickel less than copper less than zinc, will be right, as I have drawn the size you can make out. So therefore you see this first one is correct one. Negative electron gain enthalpy. Electron gain enthalpy has variation from second period to third period in the case of p-block elements, just like we know that in the case of fluorine and chlorine. Here in the case of oxygen and sulfur also it is having same discussion. Oxygen will have less electron affinity than what you can say sulfur. This is what is the reason? Same reason in which fluorine is having less value than chlorine. Due to smaller size in oxygen and fluorine, the incoming electron will have lot of repulsions. Therefore, they have smaller electron affinity. Otherwise, in general, electron affinity decreases down the group. So, in the case of this one, it should be sulfur more than selenium, tellurium. Oxygen should have been higher like this, but because of smaller size and repulsions, oxygen in fact is found to be lower than these members. So, therefore, sulfur more than what you can say oxygen, electron negativity is right, but oxygen more than selenium is wrong. So, the incorrect condition in this case is second one. Coming to the electronegativity. Electronegativity always have a tendency to increase across the period. So, if you see boron, carbon, nitrogen, oxygen, fluorine, across the period electronegativity increases. So, here he has given, of course, in the decreasing order, oxygen will be higher than nitrogen than carbon. So, therefore, this is a correct statement. And size of atom will be always higher than its cation. As the electron keep on losing, the nuclear charge increases, it will try to pull what you can say the remaining electron cloud inside. So always an atom will have more what you can say size when compared to its cation, and greater the charge on the cation, lesser will be its size. Therefore this is also right. So which is incorrect? That is in the case of second one. So you should be able to understand this part. There is a difference. I told you oxygen is lowest, but it's not higher than selenium. Now coming to the question which you have in the case of equilibrium, it is a simple one. Here first equilibrium is given, equilibrium constant mentioned as K1, second equilibrium K2. From K1 and K2 we should be able to get what you can say K3 here, because most of this relationship they are accounted for K3. So what you will see, to get K3 you are having this is 4A and here it is 2A. So what is the best way to do is to multiply the first equation with 2 such that it becomes what you can say 4A. It is on the left hand side 4A and this becomes 2B. So we will be writing this is 4A, this becomes what you can say 2B, and this will be what you can say K1 square, as I have multiplied with the component 2. How do I know I have to multiply? Because this is the desired equation, and for desired equation K3 I have to account. So therefore if I multiply I will get the entity. 4A is there in this case. Here it is C gives 2B, and here I have C as a product, but in the second equation C is a reactant. So I have to reverse this equation. Once I reverse the equation, the equilibrium constant gets what you can say reverse. As you show the representation like this, this is 2B gives what you can say C, so it becomes 1 by K2. So if you take what you can say these two values, you can cancel here and you will get the desired equation, that is 4A gives what you can say C. And this will be accounting for K3. Now K3 can also be represented here. This is K1 square and this is by K2. So therefore you will have this condition K1 square by K2. The correct answer in this case is fourth one. So always you have to get the desired equation, equilibrium constant from the given data. Either you can reverse it, multiply, all these are feasible. So coming to the next one, pH of 10 power minus 8 m HCl. This is more common what you can say question. Some of the students usually tend to keep the answer directly also. This is an acid solution. Though it is given, you should never what you can say go for a general calculation like pH is equal to negative log what you can say H plus straight away. You should not take and write this value pH as 8. This becomes a blunder. Now that is a catch in the question. Here what you have is, this is an acid solution which is not supposed to have value more than 7. If nothing is mentioned, you have to always take it as 25 degrees centigrade. Now what is the catch in the question is, the HCl solution in this case is extremely weak. So we need to add the H plus concentration of the water also. So therefore total H plus concentration, if you take total, that will be H plus concentration of acid, that is H plus concentration of acid plus H plus concentration of water. So therefore if you take H plus concentration of acid 10 power of minus 8, this is 10 power of minus 7. So the mathematical solution further can be done here. I am taking 10 power of minus 7 as common factor in these two. So therefore I will be writing 10 power of minus 1 plus 1. So therefore this is 1.1 into 10 power of minus 7. Now this is total H plus concentration of the given acid. Now you are finding out pH for the total H plus ion concentration. So pH here will be represented here as negative log 1.1 in 10 power of minus 7. If you further write this, it can be represented as 7 minus log 1.1. Even if the value 1.1 log is not given, what is the smart way of thinking is, log 1.1 is some value. So some value deducted from 7, it will be less than 7. That is the importance you have to understand. So here among these conditions, it will be slightly less than 1 because log 1 is 0, log 1.1 is a very slight value. So less than 7 it could be 6 also, but the point is log 1.1 will be very less value. So in that context we can go for what you can say 6.96 even if log value is not mentioned. Okay, that's 2. Next one, spontaneous reaction. Now thermodynamics point of view, this is a very important concept. You cannot never miss what you can say this concept in thermodynamics. Any spontaneous process will always have what you can say the delta G value should be negative. Most of the spontaneous reactions generally tend to be exothermic in nature, though endothermic reactions can also be spontaneous. By and large, if you go by generalization, delta H is negative, that is for exothermic reaction. This is a favorable process, and increase in what you can say entropy, the randomness in any process is always favorable process. So these two are favorable process, but they are not criteria. Which is criteria? This is a criteria. There is a difference between mentioning as criteria and favorable. So we have to look for a condition where delta G is negative. So these two are straight away ruled out, or I mean to say this is straight away ruled out. These three are negative. Within that, this is ruled out because in general endothermic is not that favorable. So therefore that is also we can keep aside. Increase in entropy in all natural process, generally you see it tends to increase in entropy. So therefore one will have edge over what you can say 2. So therefore the correct answer in this case is first one. Now coming to an organic reaction, you are seeing aldehyde is what you can say getting reduced to alcohol. Aldehyde reduced to alcohol, we need to have a reducing agent. PCC and tolerance component, they are oxidizing agents. These two are reducing agents. Now here if you just see, it can be 3 or 4. Okay, so here immediately we may not come to a conclusion. Coming to the second one, acid converting into alcohol. Now in this case also these are the only two reducing agents, but here the point is acid is not what you can say interacted by sodium tetrahydroborate. Now this point sodium what you can say BH4, this is component will not be useful for esters and what you can say acid. So sodium borohydride which cannot be used for acids and esters, now this has to be reduced only by 4. So therefore this is only by 4, then this has to be by 3, because by comparing the second one we could come to a conclusion. So this can be done even with sodium borohydride and lithium aluminum hydride. So these two have been decided. Conversion of acid, what you can say aldehyde to acid, and one more is conversion of alcohol to aldehyde. Alcohol is converted into aldehyde by what you can say specific reducing agent which can only oxidize alcohol to aldehyde level, not to the acid level. So we have this PCC, this is pyridinium chlorochromate. Pyridinium chlorochromate, PDC, pyridinium dichromate, these are the two components which can convert alcohol into aldehyde, either primary alcohol to aldehyde, secondary alcohol to ketone, but doesn't oxidize to acid. So therefore in the case of this one, this has to be the first one, and aldehyde converting into acid can be even done with a mild oxidizing agents like tollens component here, therefore it has to be 2. So therefore in this case it has to be 3, 4, 2 and 1. Okay, that's why it has to be, okay, right, 3, 4, 2 and 1. So therefore that is the correct answer. Let us go to the next organic question. Cumene getting what you can say oxidized in acidic medium forming two components A and B. A upon reaction gives benzene, B on reaction with Grignard reagent gives C. So he is asking C. So we have to definitely start with the cumene. Cumene is nothing but isopropyl benzene. This is isopropyl benzene we can represent. When isopropyl benzene is subjected to oxidation, this hydrogen gets oxidized, it forms cumene hydroperoxide which can be further hydrolyzed. It can form two components, one is phenol. This is, you see, this is phenol plus it forms what you can say acetone. He is not asking any intermediate component or one which forms between these two, that is cumene hydroperoxide. Now since benzene, A on reaction with zinc gives benzene, it has to be phenol only, this cannot give benzene. So therefore this has to be A, this has to be B. So this gives benzene upon reaction with zinc, yes. So therefore this condition is right. B upon reaction with Grignard reagent. Grignard reagent has condition, we know. When it reacts with formaldehyde followed by hydrolysis, primary alcohol. Grignard reagent with aldehyde, reaction followed by hydrolysis, it gives what again secondary alcohol. And Grignard reagent upon reaction with ketones followed by hydrolysis, it gives tertiary alcohol. So you have methyl magnesium bromide, this is reacting like this, it results in the formation of what you can say MgBr, and this is one more CH3 added here which on hydrolysis will result in the formation of tertiary alcohol. So therefore in this case the correct answer here is tertiary alcohol. Brass is an alloy of, some of the times we prepare what you can see in some chapters extensively, they are very big chapters, but small topics like D and F block elements where questions very often come, the simple question they have asked, brass, bronze, all these alloys which are specifically mentioned in the textbook, you need to thoroughly go through. You can keep a reference in case you have a doubt. If you say, sir, I have a doubt, generally between brass and bronze I get confused. You have a copper is a common component in both of them, because copper is a component in many alloys. Wherever you have what you can say brass, this is what you can say S, it is having what you can say in this zinc, whereas bronze it is having what you can say tin. So therefore in the case of this condition, if you see, that is brass is nothing but zinc and what you can say copper. This S should be reference, wherever you have brass, that is S is there, that means tin is not there, you can keep zinc as a reference for, you can keep some mnemonics. I am telling you one which comes to my mind. Okay, next. Incorrect statement regarding the complex. Whenever you see any complex, you have to always look for what you can say ligand. Generally carbon ligands are stronger ligands, carbon donor ligands, then you have nitrogen donor ligand. So we can figure out that the CN is what you can say a strong ligand. Coming to the point, this is -6 and the complex is what you can say -4, we can easily conclude that iron is having a charge of +2. Iron, what is the configuration which you have in this case? Iron, this is 3d6 and 4s2. Once you have iron is in the case of +2, this is 3d6 and 4s0. So now you have this 3d6, what you can say, this is a situation of 3d6 initially for Fe+2 I am writing. In presence of Cn minus, since it is a strong ligand, it can induce a forcible pairing. It induces a forcible pairing, so this gets what you can say paired up here. So once it gets paired up, in this case, how do you see this part is, this gets what you can say paired up, you have 3 lone pairs coming. So now whatever unpaired electrons, they got forcibly paired up, and we all know that if you have unpaired electron, it is paramagnetic, else it is diamagnetic. So in this case, the complex is what you can say diamagnetic in nature. Yes, it is an inner orbital complex, yes, because you are pushing the electrons inside, so this can be used up in what you can say hybridization. It is paramagnetic in nature is a wrong statement. Is it an octahedral complex? Yes, the coordination number is 6, and in this case it has what you can say tendency to form D2sp3, so therefore it is octahedral complex. So incorrect statement in this case is paramagnetic, that is third option. Okay. Now, mass of the substance obtained at cathode by the electrolysis of brine using inert electrodes when 6.023 in 10 power of 22 electrons pass through the solution. You need to remember what is brine, first part, otherwise you cannot move an inch in this question. Most of you know, but in case you don't have idea, then you will have a lot of trouble, though the question appears very simple. Brine is nothing but saturated solution of NaCl. Saturated solution means it is definitely having NaCl and H2O. Whenever you have components NaCl and H2O, we know that the reduction potential of H plus is more than what you can say Na plus. So whenever you have a competition between two cations, whichever has higher reduction potential, it gets reduced easily. So when electrons are available, H plus will have tendency to undergo reduction. It results in the formation of H2, what you can say high priority over what you can say Na plus. We know 2 electrons in the case will always speak for 2 Faraday. So it's nothing but it is 2 into Avogadro number of what you can say electron, that is 2n electrons. So 2n electrons involved, you will get what you can say 2 grams. Okay, 2n electrons. If you have what you can say in this case 6.023 into 10 power of 22, here also this n you can write, that is Avogadro number 6.023 in 10 power of 23, then in this case you will get it as 0.1 grams. You can calculate, you can of course substitute the n value, this is a very simple one. So this 2 into 6.023 in 10 power of 23 grams, you will get 2 grams. 6.023 in 10 power of 22, it comes to be 0.1 grams. We should never go for what you can say sodium, because here there is one logical point we should not miss. There are 2 in fact, but this many of the students know, there is no doubt about it. But here you should be able to understand H plus has high priority over Na plus, therefore reduction of H plus will occur. So it is, fourth option is the correct one. Next, percentage of nitrogen, this is a statement type of question. Percentage of nitrogen in nitrobenzene can be estimated by Geldahl's method. We all know Dumas method nitrogen is estimated as N2 and Geldahl's method it is estimated as ammonia. But there are some components or some specific compounds which are not suitable with respect to Geldahl's method. So Geldahl's method is not suitable for those which are heterocyclic compounds. Heterocyclic, like you will have in the case of pyrrole, pyridine, which are having nitrogen in it. It is also not suitable for those which are actually nitro compounds, NO2. Okay, it's not suitable for azo compounds, heterocyclic, and what you can say nitro. So therefore, percentage of nitrogen in nitrobenzene can be estimated by Geldahl's method is incorrect. So some of the times the theory will be very simple, but how specifically you remember that very clearly, because they have emphasized the 3 components: heterocyclic compounds, nitro compounds, and diazo type of compounds. They cannot be used to estimate by what you can say Geldahl's method. Okay, that is first part, so that is incorrect. In paper chromatography, mobile phase is liquid. Paper chromatography is a partition chromatography where you will have stationary phase where you have water molecules present on the filter paper and the solvent moving on, what you can say that. So here the components are both liquid, so therefore this condition is right. In paper chromatography, both the components which you see, stationary and mobile phase, are found to be liquid. So therefore first statement is incorrect and second statement is correct. Therefore the correct answer in this case is second one. Assertion and reasoning type of question. Reactivity of following bromides. This is from haloalkanes and haloarenes. SN1 reaction is always, what you can say, a two-step process. First step involves cation formation. You are seeing this is Rx. First step involves the cation formation, which is very difficult and it is a rate determining step. Second step is already R plus is formed immediately. What you can say, any nucleophile, even if it is weak also, it finally result in the formation of substituted product, that is SN1. So the key issue in this case is all about R plus. Whoever can form the R plus more stable, such type of halide will tend to, what you can say, undergo reaction faster. So here they have given, what you can say, tertiary halide, secondary halide and primary halide. That is order of reactivity. And SN1 is banking on what concept? The stability of, what you can say, cation. So greater the stability of cation, higher will be the reactivity of SN1 reaction. It is very strongly supporting, what you can say, assertion. So both the statements are in fact right, and the reason is supporting, what you can say, assertion, because greater the stability of cation, and we know the stability of cation is what? Tertiary is more than secondary, more than primary. Okay. So both are true and R is the correct explanation of, that is assertion. Of course, that is next. In 3D series elements, element with the least enthalpy of atomization. In the graphical condition, general properties they have clearly given with respect to graph. If you can possibly remember the values, it's fine, but it is less likely that during the exam tension and all, if you remember the values, it's very less likely. But graphical conditions, if you just see, they have given very clear graphical conditions. Starting with scandium, you have titanium, you have vanadium, then it starts, what you can say, declining. You see, this is the pattern in which you see. Okay, all these elements are placed here. Enthalpy of atomization, which is having the highest value in this case, is vanadium. So it goes, what you can say, down, and which will have lowest value in this case, zinc. So therefore the 3D series elements with a high enthalpy of atomization would be vanadium, low will be, what you can say, zinc. In this particular case they have asked least enthalpy of atomization, so zinc is the correct answer. So this question is from the biomolecules. Okay, which of the following statements regarding alpha amino acids are correct? Biomolecules is a very small topic, but certainly you will get one question. You don't have much of the numericals. If you go through the theory thoroughly and make the list of important points, you can easily, what you can say, get one question answered thoroughly. Proteins on hydrolysis gives only alpha amino acids. This is a correct statement because alpha amino acids are combining, they form polyamides, polypeptides, then finally, what you can say, proteins. GLN stands for glutamic acid. It is bit, what you can say, very harsh, but still sometimes even if you know 3 points, but he may catch you very strongly at one point. There are symbols given for each amino acids. 3-letter, what you can say, abbreviated versions are given, and here this is a close call, GLN. Usually abbreviated forms, all the letters should be reflecting. Here I am just saying one catch, GLN, GL is there but there is no N in this. Okay, I am just telling you what could be the reference. GLN stands for, what you can say, glutamine. Glutamine, where N is certainly there. So I am just telling you, in case you have looking at abbreviation, you also look into such type of smaller things so that it helps you to understand in a better way. In the case of this one, the GLU itself is, what you can say, it's the abbreviated version. So GLU stands for glutamic acid but not GLN. So therefore it is an incorrect statement. Okay. Now, amino acids with equal number of amino groups and carboxyl groups are neutral amino acids. Yes. Whenever you have in nitrogen or basic groups are more, they come as basic amino acids. If you have 2 acidic groups, it comes as acidic amino acids, like you have aspartic acid, glutamic acid. In the case of these members, when you have one amino group and one carboxyl group, they are certainly what you can say neutral amino acids. Like you have glycine, alanine, phenylalanine, so many neutral amino acids are there. All naturally occurring alpha amino acids are optically active. This point is generally taught without discussing this point. They never what you can say teach anywhere. If you see amino acids, alpha amino acids, they are all optically active except the first member, that is glycine. Okay, so this is not an issue, but in this question I feel that this is bit challenging. What happens, though you know this three, but second one is still what you can say will haunt you, some cases like that. So thorough in textbook is very important. So glycine is amino acid which does not have any asymmetry. So in that context, or in simple case we say there is no chiral center. So therefore first what you can say statement, third statement and fourth statement, if you just see, second statement is wrong and fourth statement is wrong. Okay, and he is asking the correct statement. So correct statements are first one and third one. So therefore this is the correct answer. Okay, now 1.8 grams of glucose is dissolved in 100 ml of solution. This solution is isotonic with 50% ionized exmolar NaCl. Now this question is very beautifully framed question. It's a very good question. I liked this question actually because it's looking simple, but it's not complicated. It is still simple, but you have to understand the point which is specifically mentioned here, that is isotonic. Isotonic means in general equal concentration, provided if both are non-electrolytes. If they are electrolytes, what you see, this should be the condition. This should be the condition if they are electrolytes. Okay. Now what happens, I factor? I factor for glucose, we know that it is not having any dissociation, so therefore it becomes 1. I factor we have to find out for NaCl. We know that NaCl undergoes dissociation. NaCl, once it undergoes dissociation, it results in the formation of Na plus and Cl minus. And number of particles which you are getting in this case, n value is equal to 2. And he is saying 50% dissociation. So what is the alpha value? Alpha value is 0.5. And we know the expression alpha is equal to I minus 1, n minus 1. So n is equal to 2, so this value becomes 1. Alpha is given as 0.5, so therefore I value in this case becomes 1.5. And what is the concentration in the case of glucose given? We have mentioned only the weight, so therefore we have to find out. This I1 value is 1 and find out the molarity of glucose given weight by molecular weight, that is into how much ml he has given, that is 100. So therefore it is 100 ml. And what is the I value in this case? 1.5. You find out the concentration in this case. You just work out this calculation. Okay, in this case if you work out this calculation, the C2 value in this case will be coming what you can say as the first one. Okay, just for a mathematical part I am avoiding, you can also cross check. Okay, it's very simple calculation also. It's 1.8, 180, you can understand divisibility, that won't be an issue. But here this you have to understand, this is the important part in this question. Isotonic, one is non-electrolyte, so I1 value becomes 1 and another is electrolyte, so find out how many particles are there. Okay, that's a calculated value I have written. Now in this question they have given what is the basic nature, what is the acidic nature order, and one more is reactivity towards nucleophilic addition. 3 different. Now what happens in grand test, different chapters they can club in the form of one question. That's why this is a grand test type of question. Now you see this Kb value. Whenever you have methyls and ethyls, we know that Kb value is more for secondary. Okay, more for secondary. Secondary will be more than what you can say tertiary, more than primary if the component is ethyl. Secondary is more than primary, more than ethyl if the component or alkyl group is methyl. So in this case he has given methyl only. So what he is saying, secondary is more than primary than tertiary. Of course I have told you also with ethyl, the reason is usually this is discussed with methyl or ethyl. So in that context you have what you can say this question. Coming to the Ka value, formic acid and acetic acid. Formic acid is higher value because acetic acid you have plus I effect will decrease what you can say the stability of what you can say this carboxylate ion, CH3 plus I effect. And benzoic acid, of course, this comparison is purely with respect to the Ka values, but these two can be compared very easily among the monocarboxylic acids, unsubstituted forms, simple monocarboxylic acids. Formic acid is more acidic. As the carbon chain increases, acidic character decreases. How do you compare benzoic acid? Those Ka values are given. So it's always, this is a very important textbook. They have given what you can say Ka values. Very limited discussion is there, so we have to see that meticulously. Formic acid is definitely more acidic than acetic acid, but it's more than benzoic acid or not, it's based on the Ka values. So this is also correct statement. This is also correct statement. Nucleophilic addition. What is the nucleophilic addition? In this, both nucleophile and electrophile both are added. Addition reaction in which nucleophile is first added is called as nucleophilic addition. Once it is added, this goes what you can say O minus, then H plus will attach to this, or electrophile gets attached to this. Nucleophilic addition, nucleophile, electrophile both are added. Electrophilic addition, electrophile, nucleophile both are added. But any addition reactions in which electrophile is first added followed by nucleophile, they are called electrophilic addition. Nucleophilic addition means first nucleophile is added followed by electrophile. So now here the carbon in the case of formaldehyde is having the positive charge which is not much compensated by the neighboring what you can say attached hydrogens. Whereas if you see in the case of what you can say ethyl groups and two methyls, now the very instinct of this addition is derived by the positive charge on the carbon. Greater the positive charge on the carbon, greater will be the tendency for a nucleophile to get attached to this C plus. Now coming to these two discussions, if you see here there is what you can say ethyl, CH3, CH2, C double bond OH. Now the positive charge in this case is compensated by ethyl. So therefore it is not that positive as the electrophilic carbon which you see in the case of formaldehyde. So therefore nucleophile can easily add to the carbon of formaldehyde rather than carbon of any other aldehyde unsubstituted form. So formaldehyde is more what you can say reactive than acetaldehyde, then propanol, butanol and so on. In general, aldehydes are more reactive than ketones because in ketones there are two groups which can actually compensate I mean to say it is reducing the positive charge on this carbon. If the carbon positive charge keep on decreasing, the nucleophilic addition to such type of carbon becomes weak. So therefore formaldehyde or any aldehydes generally reactive more than ketones. So in this context they have given formaldehyde more reactive than, this is propanol, both are aldehydes more than a ketone. So this is also a correct statement. So in this case all the three are what you can say correct statements, correct combination, that is okay, that's fine. Now there are two ways of seeing this. One is whichever you have in the textbook, it is generally better to completely go through that clearly. And here this is one of such case which you see in the case of coordination compounds. Here what is the logic in this case is nickel is having a stronger ligand, ethylene diamine. Ethylene diamine is a stronger ligand. Stronger ligand will have a stronger split. So when electron what you can say is moving, it will produce what you can say more energy in general. More energy always tend to give what coloration? We know WIPGR because this pattern is WIPGR. You can remember the colors directly, somebody can is smart enough to do that, it's fine. But outside what you can say, out of the box, if you just think, this is having what you can say higher energy difference. Higher energy difference we account for violet coloration. So WIPGR pattern we know, WIPGR pattern. So in this case these are not given, so we are not discussing about this. Violet, blue and green are given in this. Within green, blue they have given what you can say, one is blue, another is pale blue. Pale blue is definitely less energetic than dark blue. That's what generally this pattern wise we know that energy decreases in this order. So whenever a stronger ligand gets replaced by weaker ligands, in the case of 2H2O can replace one bidentate ligand, so this will next result in the formation of what you can say blue colored. Okay, because indigo and what you can say blue tinge can give sort of a purple color. So it is blue and what you can say indigo. Next it is having pale blue coloration. Further one more ethylene diamine bidentate ligand replaced by this. So this will account for what you can say pale blue. So therefore initially we are going this condition with first it is 2. Next what you can say you have No, I have started explanation from C. Let us come back to that one first. Now this is A, B and C I have come. Now last all ethylene diamines have been substituted with H2O, which is a weak ligand. It does not have that much of splitting, so it gives less energetic among this discussion, so therefore it becomes green. So therefore A is nothing but what you can say 4. Next B is what you can say, that is, I have told you this is what you can say 3. And coming to this one, it is having the stronger color, that is violet, and here this is what you can say green. So you can also remember, but I found this could be a logic behind this one. Okay, that's why it has emphasized and also shown with the colors. You can also figure out that. Okay, here this. So answer in this case is 4321. That's right. 16 grams of methane. Now coming to stoichiometry based question. 16 grams of methane on combustion with 70 grams of oxygen gives V liters of CO2 at STP. Value of V is. Sometimes examiner also give you some values which are not needed, only to ensure that you don't get carried away by such type of values which are unnecessarily given more quantity. So it is something like 16 grams of methane. Let us write the combustion part. In this case, you know, methane on combustion does result in the formation of CO2 and what you can say H2. 16 grams of methane requires mere 32 grams of what you can say oxygen. So this is into 2. This is 32 grams into 2, that is 64 grams. He has given already 70 grams. That means here 6 grams are what you can say excess. So this is an excess agent. This is a limiting agent. So this will react completely. And once it reacts completely, it is 1 mole to 1 mole relationship. It forms 22.4. Okay. I think less number of students can pack here. It's a straightforward question. But only thing is, we should not take the count with oxygen. Like what I mean to say, 64 grams gives 22.4 liters, 70 grams gives how much? Don't do this calculation because that is an excess agent. In qualitative analysis, which of the following is used in the conformation of ammonium ion? Now we know group cations, one which comes in the last, that is NH4 plus. NH4 plus is specifically what you can say determined by Nestle's reagent, that is K2HgI4, okay, which gives what you can say reddish brown color. Okay, this is a reddish brown color, that is a characteristic coloration for what you can say this cation. In practical chemistry, remember most important things, that's what you can say enough, we need not have to go more details about that. In practical chemistry, we know that ammonium ion can be detected with Nestle's reagent, this is K2HgI4. So therefore the correct answer in this case is first one. Of course we know Lucas reagent is for alcohols, Hinsberg reagent, it is nothing but we use what you can see in the case of amines, segregation of amine, Tollens reagent. So you can certainly know in these three are absolutely cannot be for ammonium ion. So worst case, nobody can do even wrong answer, that is Nestle's reagent, because these three cannot be, because they are organic reagents not used for ammonium ion at all. So examiner has given a very friendly paper and a slightly tricky paper, we should appreciate that. Okay, it's from Sri Chaitanya. Now next one, for the titration of HCl versus a weak component, that is sodium carbonate, the indicator used is. Now we know that starch is used for what you can say wherever you have iodine is involved, so therefore in this case it cannot be. Phenolphthalein is used for stronger bases. Sodium carbonate solution is not a stronger base. Phenolphthalein cannot be used in the case of what you can say weak base components. Litmus is more what you can say the values with reference to 7, so therefore we don't use. And methyl orange, methyl red are some components which can be used for strong acid versus weak base titrations. So therefore the correct answer in this case is methyl orange. Titrations, that's why practical chemistry, just go through briefly all important points you underline and remember, don't go too deep into that one, just group cations, important what you can say identifications, important conformations, certainly go through. Practical chemistry questions will certainly come. 13th group, the element with least melting point is. The melting point in the case of these members, these are all solids, we don't have any gaseous member. Boron is having highest value because in this case it is having the crystalline structures. So boron is higher than aluminium, which is having what you can say higher. Gallium, we know that it has less melting point. It melts at room temperature. In summer it melts at room temperature. We also call it as summer liquid. Cesium and gallium, why you call summer liquids? Because in summer if you just take in the palm also, the temperature is enough to what you can say melt them. So therefore gallium has low what you can say melting point. Then it is indium and thallium. So in third group, remember, boron, aluminium, gallium, gallium it decreases and till thallium it increases. If you see nitrogen family, it is actually reverse. Nitrogen, phosphorus, arsenic, arsenic increases because of the layer structure. Of course, that is with respect to fifth group, but here you remember this. So this is having low melting point and very high boiling point, nearly 2000 degrees. Therefore, it is also used in high temperature thermometers. Okay, high temperature thermometers. So that's the hydrate of 17th group with highest melting points. Now boiling point generally you can answer with hydrogen bonding. Melting points, because table is given, and once such type of questions will come, if you don't remember, you cannot work out anything and get an answer here. This type of things are very tricky. So therefore what you'll see that here we have the condition something like this: HF, HCl, HBr, HI. As a molecular weight increases, generally tend to increase, but HF is not found to be lowest. Its value is what you can say found to be higher than HCl and HBr. So the correct order in this case is HCl, HBr, HI, HF and HI. This is melting point of view. Same thing if you see boiling point of view, then also HF will have a greater leap, and here you will see HCl, HBr, HI, then HF. So boiling point of view, it has a greater leap. So therefore HF will have higher than HI, but melting point of view, we just see the HF will be higher than HCl and HBr, but not highest. So therefore HI is the correct answer in this case, because he is asking highest melting point. Okay, next. Which of the following on ozonolysis in presence of zinc H2O gives same product? Ozonolysis has a logic. Anywhere, what you can say, whenever you come across double bonded, triple bonded conditions, we know ozonolysis will have tendency to break the bond. So this is a C2 carbon, this is C3 carbons, it cannot be the same product. Here of course you see this is a component something like C3 system, this is 1, 2, 3, 4, this is C4 system, it cannot be the same product. It is also asymmetrical in this case, this is C2 and this is C1, and in this particular case ozonolysis break the bond and add oxygen. Here you see this is both case you will get acetone, which is what you can say same product. So the correct answer in this case is fourth one. Okay. Next. Only small amount of HCl is required in the reduction. This is a statement type questions, not assertion reasoning. Reduction of nitro compounds with iron scrap and HCl in presence of steam. Now whenever nitro compounds, let us take the discussion of nitrobenzene reduction. If you just see this is iron and HCl, this becomes what you can say, of course this is aniline. Now what he is saying, you are taking less amount of HCl for two reasons. One is, if you take excess and aniline forms, this is a base. This base what you can say react with acid. So therefore in that context also we have to take smaller amount. So smaller amount of HCl taken is very important. Second thing, there is one more what you can say understanding from this. If I take smaller amount of HCl, initially we use this as FeCl2 and H2. This H2 is used for the reduction in this reaction, but the FeCl2 which you are seeing is also subjected to hydrolysis. This results in the formation of, this is HCl. So it is again acting as a source of what you can say. So whatever FeCl2 form which hydrolyzes to give HCl, which can be reused. Okay. So what is the point in this case is, they have given under the nitro compounds reduction to amines, only small amount of HCl is required in the reduction of nitro compounds with iron scrap and HCl in presence of steam. This condition is absolutely right. Okay, so therefore this usage of this reagent is absolutely right. Small amount is for two reasons. Excess can again further react with base, so we cannot take the excess amount. Second point is, the HCl reacting to form FeCl2 on hydrolysis gives back HCl. So therefore FeCl2 on hydrolysis gives HCl. So both the statements are what you can say correct in this case. So statement 1 and statement 2 both are what you can say correct. Number of radial nodes maximum. You know, number of radial nodes can be determined from the expression n minus L minus 1 radial nodes. So in the case of first one, let us apply in the case of first one. Now that is principal quantum number is given as n and f value. L is equal to what you can say 0, that is S. L is equal to 1, that is P. L is equal to 2, that is D. L is equal to 3, that is F. So therefore in this case it is 3, what you can say, this is how much you will get in this case, the value is 1. Okay, the number of radial nodes are maximum in. So let us see in this case, this is first one. For 1s orbital, this is 1 minus, this is 0 minus 1, so this becomes 0. For 3d orbital, this is 3 minus, what is the d value in this case, 2, so it is minus, so this is also 0. Coming to the fourth one, 2p, for p L value is 1, so it is 2 minus 1 minus 1, so 0. Therefore the correct answer in this case is first one. So Lewis structures, whenever they are given in chemical bonding, you have to remember, because at this juncture most of the questions can be solved or remembered, but some of them are like this type of things, unless you know the structure, whether they have sigma bonds or pi bonds. That's why in chemical bonding, the structural part of ions and molecules, you should have complete idea, especially those which are mentioned in the textbook, first point. Those which are out of the NCERT, even if you don't remember, it's not an issue, don't take any strain. This is NO2 minus. NO2 minus, you have to remember what you can say, this is negative charge. So wherever it is having pi bonds and sigma bonds, it has generally tendency to what you can say undergo resonance. You have CO2, this you have to remember like this. So sigma bond, pi bonds, we know the bond order also calculation, we write the structures of resonance. Coming to NO3 minus, NO3 minus, here you will have the condition like this. Sigma bond and pi bonds are there. And in carbon dioxide also, these are, you have, this is not having charge, but you can also write what you can see in the charged forms. Okay, this is something like this. Okay, so whenever you write like this, it is a charged form, uncharged, both are resonating structure. This is more stable than that. As always we have the condition. So all of these have the tendency to show resonance. Okay, so this 3 are ions given, this is not given in ion. That can be a trick, and ABC is also given. So don't get carried away. In resonance we all learn the charged forms, uncharged forms, this is more stable than this. So all the things, apply the concept. You also look whether ABCD is there or not, give a thinking and then rush, because this type of things look simple, but it can trick you, what you can say. Which of the following reaction has delta H more than delta U? Let us see what is the relationship in thermodynamics we study in the case of delta H, what you can say, and delta U. This is delta N represents number of gaseous products minus number of gaseous reactants. If the delta N value happens to be more than 0, then obviously delta H will be more than what you can say, delta U. Okay, so here number of moles, in this case it is 1, this is what you can say 1. So therefore products minus reactants, which is the calculation for delta N, in this first case it is 0. Here it is 1 particle and this is 3 particles. So it is 1 minus 3, therefore it is minus 2. In the third case, there is only a gaseous component of product here. There is nothing in the reactants. So it is 1 minus 0, therefore it is 1. Okay, that is a positive value. Let us see in the last one. They are all aqueous or what you can say solid components. They are not the gaseous products or reactants. So in this case, it is what you can say 0. So in which case delta H will be more than what you can say delta U will be in the case of third one, because delta N is positive in that case. Rest is either negative or 0. The aqueous solution with maximum pH at 298 Kelvin is. This question has to be understood based on the hydrolysis concept. Strong acids, strong base salts, they do not tend to undergo hydrolysis. Okay, they undergo ionization. For example, you have potassium sulfate. It can undergo ionization, but the conjugates which are coming from stronger acid and base, they will be weak. They are weak enough what? They don't react with water at all. Coming to the NaCN, NaCN is giving two ions, Na plus and Cn minus. It is coming from NaOH, therefore it is weak conjugate. This is coming from weak acid HCN, so therefore it is a strong conjugate. Whenever anionic hydrolysis occurs, it produces anion. And what is anion which can result from water? OH minus. So this solution will be basic. So in this case, what you can say, pH will be more than 7. Over here pH will be equal to 7. Coming to this one, this is a negative charge coming from sulfuric acid, this is plus 2. So since this is coming from sulfuric acid, which is a strong acid, the sulfate ion is a weak conjugate. Whereas copper plus 2, it's coming from base which is a weak base. Copper plus 2 is what you can say, a strong conjugate. Whenever it reacts with water, it produces H plus. So pH will be less than 7, so it cannot be maximum. And this is straight away an acid. We need not have to discuss about cationic or anionic hydrolysis concept. HCl being what you can say acid, pH will be less than 7. So there are two cases where pH is less than 7, one is equal to 7. There is only one case where it is more, so that will be maximum pH. EMF of the cell. We know this concept is from electrochemistry. We can apply Nernst equation where he has mentioned magnesium is getting oxidized, and left hand side, right hand side has been clearly given. It's a simple concept, you see. This is E is equal to E naught cell minus 0.0591 by, this is what you can say here, let us say number of electrons, log, what is formed on the product side is Mg plus 2, so therefore it is Mg plus 2. Since magnesium is losing 2 electrons, we can straight away say N is equal to 2 here, and silver Ag plus, it is having what you can say taken 2 times, because we can write this reaction very simply like this. Magnesium is above what you can say hydrogen, so it will tend to undergo oxidation. This is plus 2, and Ag will now what you can say combine with 2 electrons and forms what you can say Ag. So therefore the ionic form product, that is magnesium, product by reactant. So in this case also. Now if you check the value, this is 10 power of minus 2, this is 10 power of minus 1. This is 10 power of minus 2, this is 10 power of minus 1 to the power 2. So now what happens? This is 10 power of minus 2, that is 10 power of minus 2. This gets cancelled, it becomes log 1. Log 1 is 0, so this entire entity becomes 0. So whatever E naught cell is given, that itself becomes what you can say electrode potential value. So therefore 3.17 which is given E cell, that itself is what you can say the electrode potential in this case, because the concentrations are what you can say cancelled in such a way, it result in log 1 value. So therefore the correct answer is fourth one. Coming to the chemical kinetics question, it is assertion reasoning question. Rate of what you can say reaction increases with temperature. Okay, rate of reaction we generally know increases with temperature, because whenever you are heating, the kinetic energy of the molecules will increase, and the collision chance what you can say increases, collision frequency also increases. Whenever overall collision frequency increases, definitely fruitful collisions also increases. So first statement is what you can say assertion is correct. At higher temperature, the fraction of molecules with higher kinetic energy increases, and thus increase number of activated collision molecules. Activated molecules will have fruitful collisions. So both the statements are absolutely right, and R is perfectly supporting the first one, because whenever you have more kinetic energy, they are more likely to what you can say collide very effectively. Both what you can say direction is there as well as energy. That means there is more probability. So therefore first one is correct answer in this. This question, he has given what you can say a graphical form, rate constant of a first order reaction. There is no need to panic in this question, though the graphical condition looks where to start from, you straight away write a first order reaction. This is 2.303, this is what you can say T log A by A minus X. What is A? Initial concentration. Initial concentration, you can say this is initial concentration when time is equal to 0. And in the graph, where is the time? In this case it is 0, this is 0. So this 0.602 is initial concentration. And after a time period, how much it is? 136. What is the concentration? A minus X. So therefore A you substitute here, that is straight away you can see K is equal to 2.303 by log, what is the A value? When time is equal to 0, this is 0.602. What is a minus x? This is 0.301. What is the time period here? 136.8. Now if you observe this value, a and a minus x is exactly half of this. 0.602, if you make half the concentration, 0.301. That means here you should understand T is nothing but T half in this case. So whatever T is given here, it's nothing but straight away T half. So in the problem, I am just writing now T half, though it doesn't mean T half. Based on this question, as a precursor, I am just writing like this, because this concentration has become exactly half, if you can figure out. Okay. Now, this is what you can say T half. Let us write, I am writing like this. So this goes what you can exactly 2 times 0.3010 again, log 2. So when you multiply with this, it becomes 0.693. And what is the value here? It is given 136.8. So this comes as 0.005. So it comes as 5 into 10 power of minus 3. Correlation part you can do more better than me, but here you have to understand this part. Though the question is given in the graphical form, it is the easiest question presented in a very beautiful way. I highly appreciate what you can say this type of questions, those students who have actually done it. Okay, it's a very simple concept. You should not get scared. You should understand the logic in this. Okay, next. Read the following. Some of the conditions which you have in the case of P block elements, we have a limited syllabus. So every para, every corner, you just go what you can say through thoroughly, so that you don't miss any point, because this type of question, if you don't remember, you will be helpless, or you should be very smart enough to understand from the options. Read the following. He is asking incorrect statements. Let us see one by one. RnF2 has been what you can say isolated. Now what you have to see in the case of this one, he is asking incorrect statements. RnF2, if you see one of the conditions they have given, it can be what you can say identified, but cannot be isolated. It's given clearly in the case of what you can say the noble gases. RnF2, it is can be identified but cannot be isolated. Natural gas is a commercial source of what you can say helium. We know natural gas contain what you can say helium. So it's a correct statement. First one is a wrong statement. Okay, this of course remembering RnF2 is a bit challenging, I do agree, but that's the way the questions are. You may have hardly one or max 2 questions about like that. The only noble gas with positive electron gain enthalpy is helium. This is wrong, because we know that all noble gases have stable configuration and all of them have what you can say positive values. They have different positive values. Neon what you can say has highest value anyhow, but it's not the only element helium. All the members have what you can say positive value. Coming to xenon oxidifluoride has T-shaped. Now we have this condition xenon, which if you write the structure, it is having what you can say 8th group element. It has what you can say 8 electrons, 4 are involved in bonding, 2 are present like this. So it is having perfectly what you can say T shape. So B and D are correct statement, but in this question is asking incorrect statements, so which are A and C. Oxidation state of europium and cerium in aqueous solutions are respectively. In lanthanides, they always mention in the table they have given, in the points they have mentioned that +3 is a common oxidation state of lanthanides, +2 is a common oxidation state in the case of D block elements, because very commonly you see +2. Here you have to understand cerium is having configuration what you can say with +4, which is equal to what you can say xenon configuration, that is 54, which is equal to xenon. But even though it has a stable noble gas configuration, it is found to be a stronger oxidizing agent, because it undergoes readily reduction to form what you can say cerium +3. Now this is a very important point to understand why they say common oxidation state is +3. Cerium +4 has a value 54, cerium is 58. After losing 4 electron becomes cerium +4, but even then it has xenon configuration. It gains 1 electron and convert to what you can say cerium +3. That shows that +3 is so common that even highly stable one which has xenon configuration also is converting into +3. Europium, this is +2. it will lose what you can say electron. In this case, will have what you can say this and attain what you can say +3. It has a stable configuration, but still it loses one electron because +3 emphasis is so high in these members. Even if they have stability individually, they still tend to attain the +3 form either by gaining electron or by losing electron. So +3 is a common oxidation state that can be presented in this form. So keep an eye on what you can say the configurations and stability part. Oxidation state of central metal in Wilkinson catalyst. Even if you don't know about what is Wilkinson catalyst, also not an issue. You don't know anything about rhodium, also not an issue. This is looking like phosphine to you. This is pH3. Okay, pH3 is something like neutral molecule. Okay, just you see if you answer this with calmness, you can easily get it, pH3. In this case, it is instead of each H, you have phenyl rings. Otherwise, there is no big difference. So it is a neutral, this is also neutral. So it is not contributing anything to what you can say, the charge aspects of what you can say rhodium. Only component which you have is chlorine here. So chlorine being -1, rhodium has to be +1. So even if you don't know anything about rhodium, which group, whatever it is, it's not a big issue. Based on Cl-, you have to catch this rhodium is +1. And where the instigation should come is, you should able to figure out that it's looking like phosphine. So it is a triphenyl phosphine is also neutral ligand, just like what you can say phosphine. So therefore +1 is the correct answer. Coming to the next question, freezing point of 0.2 molar glucose is lesser than 0.1 molar urea solution. Freezing point is depending upon the number of particles, it definitely get influenced. If you have more particles, you have more depression in the freezing point, so freezing point will be lesser. You have more particles in the case of this because both are non-electrolytes. This is 0.2, this is 0.1. Freezing point is less. Yes, when you have more particles, depression in freezing point will be what you can say more. So it will be more in the case of glucose because it has 0.2 moles per liter. That's what it has when compared to urea. So therefore this statement is correct statement. So this statement is what you can see, we have this correct statement. Coming to the concept, molar mass of glucose is more than molar mass of urea. Molar mass of glucose is 180 and urea is what you can 60. So there is no big issue in understanding this part. This is also right. But what you have to understand is molar mass is not needed because already number of moles are given. You are not finding out anything with the molar mass. Suppose weights are given, you figure out glucose happen to have more moles. It could have been close. It could have been an explanation. That's a different issue. But in this case, this is individual entity which is not much related to this. So both are what you can say correct statement, but it's not an explanation. Explanation you can say the 0.2 molar glucose has more number of particles than 0.1 mole of molar what you can say urea. So that will be what you can say condition. Which of the following oxides is used as bleaching agent for paper, pulp, textiles and water treatment? This type of questions I can understand your feelings. All these are very good oxidizing agents. They are highly reactive. This type of important concept you learn. All these are oxidizing agents. They are important. What you can say, they are highly reactive. They have explosive nature. All these are generally we come across more in the discussion. But among these members, you have ClO2 is having the bleaching nature. And it is used for bleaching what you can say paper, pulp, textile and water treatment, which is given in the textbook NCERT. So just see this part. Otherwise logical point of view, there is nothing like we have to evaluate something from the question. You have to remember this is a purely memory based question. Okay, next. Stability of first cation, of course this is statement type, not assertion reasoning. This neutral molecule, perhaps I can represent this is CH2, this is CH3. So yeah, this is CH2 double bond, CHC double bond OH. Because based on the resonance pattern we can understand this. If these electrons go here, this becomes minus. That will be the general pattern. So in the first case, this is the negative charge, here positive charge. In resonance, what you have to always remember that positive charge is there. Usually there is a shortage of octet. Generally we will have what you can say less stability. But in this structure, I have a positive charge on carbon as well as positive charge on oxygen. Don't get carried away by seeing straight away the carbon having positive charge, because some of the students will remember less than octet it is less stable. Yes, but here this is less than what you can say 8 electrons, oxygen is also less than 8 electrons. Okay, this is the important part in these two structures. Carbon with positive charge, oxygen which is more electronegative having the positive charge, there is a difference. More electronegative with positive charge will be less stable. So in this case, stability of this one is more than this is incorrect statement. It's a not correct statement. IPAC name of the structure is given. We know there is a double bond and triple bond. Whenever there is no chlorine, I would have rather gone with a double bond. But when you have what you can say substituent, you always tend to give priority to substituent also, because there is no big difference. If substituent is not there, I would have started with what you can say double bond. So in this case 3-chloro, this is 7 members, it is hept. On 6th position there is a ene, because always naming is enine, and first position there is 1-ine. So this is correct condition. Don't get prejudiced mind like double bond will always win over triple bond at equal numbering. Yes, this is one case where examiner has very properly selected a question where double bond doesn't get priority over triple bond. The type of things where it fails, you should understand from this example. Very good example. So therefore first one is incorrect, second one statement is correct. So the correct answer in this case is second one. Wrong match. Here given couple of reactions in this case and is asking which one is incorrect, what you can say condition. Because the names are given, it becomes easy to analyze. We precisely go to that equation. First one is aldol condensation. This component has alpha hydrogens, so this can involve its hydrogen, and benzaldehyde involve what you can say oxygen. In this it undergoes what you can say condensation. So therefore it results in the condensed product where double bond is found between this carbon and this carbon. So therefore first condition is right and it is a crossed aldol condensation. Second one, toluene on reaction with acetic anhydride in presence of chromium trioxide, it gives benzaldehyde. This is also absolutely right, but this is not Itard reaction. This reaction is right, but it is not Itard reaction. Itard reaction we use what you can say chromium oxychloride or chromyl oxychloride. Chromyl oxychloride is used, which is not mentioned here. So that is an important reagent we use, what you can say oxidizing agent in the case of Itard reaction. Chromyl oxychloride, which is not given in this, so it is not an Itard reaction. Next one is Hoffman's bromide, where primary amide is converted into primary amine. Primary amide is converted into primary amine with one carbon less. Hoffman bromide degradation. There is loss of one carbon, hence it's called degradation. That's right. And in the case of diazonium salt reactions, they are very susceptible or what you can say potential components to form several aromatic compounds. With copper and HBr, it results in the formation of bromobenzene. Copper and HCl, it forms chlorobenzene. So this comes under what you can say Caterman reaction. So the wrong match in this case is second one. So our answer point of view, it will be 2. Okay. Next, read the following. You have tertiary alcohols gives immediately turbidity with Lucas reagent. Tertiary alcohols, Lucas reagent is concentrated HCl plus anhydrous ZnCl2. Okay, we have ZnCl2. So therefore HCl will result in the formation of more stable cation tertiary than secondary than primary. So tertiary alcohols are more reactive than secondary alcohols than primary alcohols. So here he has given tertiary alcohol immediately give precipitate. It is a correct statement. Ethanol gives characteristic color with ceric ammonium nitrate. Most often it is represented as CAN. Okay. CAN is one of the type of reagent which gives color with alcohol and phenol. With alcohol it gives what you can say red color. With phenol it gives what you can say, some books they write violet, some books they write dark red also. You can just check that one. That is dark red is given morely what you can say in practical books. So ceric ammonium nitrate will tell you whether there is a phenolic group or alcoholic group. Ferric chloride is not like that. Ferric chloride gives coloration only with phenol but not with alcohol. So that is a important difference between ferric chloride, neutral ferric chloride and ceric ammonium nitrate. Ceric ammonium nitrate gives determination of OH group whether it is in alcohol form or phenol form. So therefore it gives characteristic color, that is also right. Phenol gives characteristic color with neutral ferric chloride. Okay, for a moment I get carried away. I told you the same point which is actually there in the third one. And benzaldehyde gives red precipitate with Fillings reagent. Fillings reagent, we have what you can say sodium hydroxide. When you have sodium hydroxide base present, and this is such type of aldehyde which is not having alpha hydrogen. So the Fillings test fails in these cases where you don't have alpha carbon and alpha carbon is there, but there is no alpha hydrogen. So whenever components aldehydes which doesn't have alpha hydrogen, they rather undergo Cannizzaro reaction, and they don't interact with what you can say Fillings component straight away, because NaOH will carry out what you can say Cannizzaro reaction by first attacking what you can say in this case OH here, then there is a hydride shift from one benzaldehyde to another benzaldehyde form. So therefore it fails in the case of aromatic aldehydes without alpha hydrogen. So therefore in the first three it is actually right, but it is incorrect in the case of fourth one. So here is asking correct statement, so it is first three are right. Coming to the estimation methods, that is from some basic principles and techniques chapter. Usually what you can see, we see Geldahl's method, Dumas method very commonly, but here they have asked what you can say estimation of phosphorus, that is by Carius method. Magnesium pyrophosphate, phosphorus is estimated in the form of magnesium pyrophosphate. If you work out what you can say the molecular weight of this component, because all are known to us, it comes as 222. Phosphorus you should take 31, and here how many phosphorus are there? 62 grams. So you should understand every 222 grams of magnesium pyrophosphate has 62 grams of phosphorus. So the percentage of phosphorus is can be written like this: 62 into how you are estimating phosphorus as magnesium pyrophosphate. So it is weight of magnesium pyrophosphate P2O7 by this is 222, and how much is this percentage in the we are calculating with respect to the weight of this weight is what organic compound. So we know the weight here which is given 0.222. Okay, examiner is very slender on you, he has given the calculation which can match with this value, and 0.12 you substitute here and just work out what you can see. In this case I believe in this as students worked out it comes to 51.66. okay, I believe that probably yeah that's right. In the conversion of phenol to salicylaldehyde, the electrophile involved is, you know, this is a very important reaction, that is Reamer-Tiemann reaction. In Reamer-Tiemann reaction we use what you can say chloroform along with what you can say NaOH with the phenol. Now the initial reaction is actually between chloroform and what you can say NaOH. Chloroform what you can say whenever it reacts in presence of base, it first tend to form CCl3 minus, and what you can see here it forms H2O. This CCl3 minus will shed what you can say the excess negative charge like this, and here it forms what you can say this dichlorocarbene as a intermediate. This start undergoing reaction with phenol, which of course so we know that it's highly electron rich and it is electrophile. What you can say this CCl2 electrophile will now come here, it will establish the CC bond formation. Of course that later part is not the part of this question. In the conversion of phenol to salicylaldehyde, it is basically a Reamer-Tiemann reaction. What are the components which used here, because phenol anyhow he has given, so I have discussed with respect to chloroform and NaOH. So the correct answer in this case is CCl2. Weight of carbon 12 which contain 1 mole of electrons is, this is a simple stoichiometric question, don't mess up in this case, just what you can say hold your nerves. This is you write properly, you know 12 grams of carbon is having, this is what you can say atomic number is 6, so 6 into what you can say n electrons, very simple, because atomic number is 6, 12 grams is 1 mole of what you can say atoms, each atom has 6 electrons, so 1 mole of atoms has 6 n electrons. Now he is asking what is the weight of C12 which contains 1 mole of electron. We know 1 mole is nothing but, this is I am writing this is 1 n electrons. So it is how much? So simple what you can say 12 by what you can say is this is 2 grams. So कितना simple question है, but presentation बढ़िया है, both beautiful question and a simple what you can say solution also. Yeah, that's right. Next, coming to one more assertion reasoning question. Nowadays iodoform is not used as antiseptic. Yeah, you have other components which have come, even sodium hypochlorite is used as what you can say important antiseptic. So earlier this iodoform was used as an antiseptic. In the slightly the presentation could have been in the form of what you can say earlier iodoform was used as antiseptic, but anyhow nowadays iodoform is not used as antiseptic. We can excuse the presentation here, so we can still take this as what you can say correct. Iodoform, it is not used as antiseptic is because iodoform has what you can say the odd smell. Okay, the smell is what you can say highly objectionable. In the textbook it has written as objectionable smell. So as an emphasis that iodoform is not being used because of its very odd smell, this condition has been discussed. So what you have here is objectionable what you can say smell is the key issue. It should be kept in your mind. Don't worry about especially this question. It could have been slightly presented in a different way. We know iodoform always acts as an antiseptic by liberation of what you can say iodine from it, not because of what you can say the iodoform itself. So that is a key issue. So here what you see that the first statement is right. Second statement, iodoform has objectionable smell, bad odor. People generally don't tend to use a bad what you can say smelling, especially kids will straight away throw it. That's the reason in that context some other what you can say antiseptic members are used. Okay, of course here it is first statement is right, second statement is wrong. It is based on what you can say textbook what you can say presentation. Coming to the organic question, which is alkyl halide reaction. This is the 90th question, so it has perhaps it is the last one. So ethyl chloride reacting with silver nitrite. Silver nitrite and potassium nitrate, both of them you know very well. They have what you can say nitrite form is an ambidentate nucleophile, but where from it is derived is very important. When it is silver nitrite, this is ethyl chloride reacting with silver nitrite, it results in the formation of nitroethane. If it is potassium, it becomes ethyl nitrite. When it is subjected to acidic reduction, we know that a nitro group, whenever it is subjected to acidic reduction, this has what you can say tendency to get reduced. It becomes, I am writing ethyl, this becomes NH2. So nitroethane tend to convert into ethylamine. He is saying the correct statements about B. So who is B here? This is ethylamine. So now B gives alkali insoluble derivative with what you can say benzene sulfonyl chloride. In the case of what you can say B, it has important reaction benzene sulfonyl chloride, that is Hinsberg reagent. It reacts with what you can say primary amine. Once it reacts with what you can say primary amine, it undergoes replacement of hydrogen. One more hydrogen is there, it becomes soluble. So in this case it is insoluble. He is saying gives what you can say insoluble derivative with benzene sulfonyl chloride. It is wrong because it is a primary amine. It gives what you can say soluble derivative. Secondary amine gives insoluble derivative. B is more basic than ethylamine, diethylamine, sorry. So diethylamine is pumping more electrons on the nitrogen. So diethylamine obviously will increase electron density on the nitrogen. So therefore this is what you can say B is more basic than diethylamine. This is B, which is more basic than diethylamine. Here it is incorrect statement. B is not what you can say more basic than that. B gives ethyl alcohol on reaction with, yeah, here it is insoluble is said, so I beg your pardon. This is also incorrect because it gives soluble derivative. I was talking about primary amine isn't, so therefore it gives a soluble derivative. Secondary amines gives insoluble derivative. Tertiary amine of course doesn't react with Hinsberg reagent. So both of them are incorrect. And the last here you just see B gives ethyl alcohol with this. This mixture will result in the formation of HNO2, and HNO2 always converts NH2 group into ethyl alcohol. This is OH. So therefore this statement is actually correct statement. B gives N-methyl ethanamide with what you can say this N-methyl ethanamide. So you are having this is the reaction in which you have the product formation is like this with he's saying acetyl chloride. So this is not N-methyl, it should be what you can say N-ethyl. So therefore it is also incorrect statement. So the correct statement in this case is third one. So this is the last one, and the correct answer in this case is 3. It's already late. I appreciate those who are present. You understand the solutions. So please be confident. Thank you.